Well, thank you so much for being here bright and early on the first day of TSC 2022. It's good to see so many people in person. The last time we had this conference here in 2020, it was just the four of us, Stuart, Abby, and myself, and the AV team in this whole hotel alone. It was very spooky. So it's so good to be here in person with all of you. And hello to everybody joining us out there on the internet as well. Apologies for the delay. We had a little bit of a technical problem, but we're good to start. So I'm Jay Sanguinetti. I'm the Assistant Director for the Center for Consciousness Studies and have been involved with ultrasonic brain stimulation for some time today. So that's the topic of this, this workshop today. And I'm going to put up a slide. Can you give me presentation mode? That'll come up in a second. So what are we doing here today? We're talking about ultrasound brain stimulation, and this is an exciting new technology that has the potential to radically alter the way we do neuroscience, and it'll allow us to address many of the deep questions in the field of consciousness studies. So this is a really exciting tool and technology for us at this conference. And the goal of the workshop today is to give you an intro and a sense of where we are with this technology currently. We have some of the founders of the ultrasound brain stimulation field on our panel. So we're very lucky today to have our speakers. And they're going to give you a sense of the history of this technology, where we currently are. And then the hope is to give you a sense of where we're going with this, and why in the consciousness studies field, particularly, we might be so excited about this technology. This field has been growing very quickly over the past 5 years, but some of our speakers have been involved in this for the better part of 20 years and have been cultivating this field for a long time. And there's just been an explosion in interest in this technology very recently. So you're going to get a sense of that, I think. And what we'll do is, as you see at the outline here, we're going to have a couple of presentations that are about 30 to 35 minutes apiece. These are going to give you a sense of how we're currently using this technology and some of the history. You're going to hear about things, for example, Seung-Schik might tell us about brain-to-brain interfacing with this technology, the use of this technology to wake people up from coma from Monti. Dr. Bystritsky is going to give us, I believe, a history of the field and kind of a projection into the future. And I'll talk a bit about our work altering consciousness in healthy subjects and trying to facilitate advanced states of meditation and mindfulness. So we'll just give you a little bit of a sampling of the field in these talks. And we will take questions after each of these. So please hold your questions till the end of each of these talks. And we'll take 5 or 10 minutes of questions apiece. Then we'd like to do a panel discussion. We might invite a few people up from the audience to discuss some of the things that we're talking about today and really dig into some of the details about the nature of this technology and really how useful is this going to be both for neuroscience, but particularly for consciousness studies. And then if there's time at the end, I know we're sort of starting a little bit late, we'd like to do a little bit of a workshop with the audience. So we'd like to know your feedback about this technology, particularly about how this could be used to address some of the deeper questions in consciousness studies. All right. So that's our outline. We'll take a couple breaks in between here as well, but we might have to cut those a little bit short. And I'll introduce the speakers very briefly here. So as you see on the screen, first up, we're going to have Seung-Schik Yu. He's calling in from Harvard University. Seung-Schik is one of the folks who's been there really at the beginning of this new field of using ultrasound as a non-invasive brain stimulation technology. He's going to give us an intro to this technology. He's been doing some very beautiful work in rodent and mice models, as well as human research. As I said, he recently did a study where he had brain-to-brain interfacing between rats and humans, transmitting visual information from the rat to the human, or from the human to the rat brain, and vice versa. Then we have Martin Monti calling in from UCLA, has been doing some really beautiful work in disorders of consciousness and and quote, waking people up from from coma, waking people up and making them conscious. So he'll give us details about that. I'm going to talk about our work here at the University of Arizona, applying this technology in healthy people. And then Dr. Bystritsky is going to give us an overview of the field. Okay. So we're going to switch to Seung-Schik's Zoom. I'll give a brief intro, very, very brief, since we're a little bit late here. Hello, we have you on the big screen.
Okay, great.
We've got you and we can hear you.
Okay, so I'm so glad to be the part of this wonderful workshop. It's also an honor for me. I'm actually here to learn about the consciousness, and I would like to get some feedback from all the audiences as well. So without a further ado, I would like to share my screen. So we are gathered here today to learn about how our brain function in terms of consciousness, and if there's any way that we can possibly modulate it using non-invasive techniques. So that's one of the things that I'll talk about. One of the topics that I'll like to discuss with you is to analyze the changes in offline resting state functional connectivity. I'm going to get to that, what it is, among humans that has been stimulated on the somatosensory circuitry by using transcranial focused ultrasound. So before that, let's go over some of the tools that we have in terms of spatial resolution and temporal resolutions. There are a variety of current tools out there. You probably heard about, you take medications, or sometimes we do brain direct stimulation. You call it microstimulation. And it all has a different merits and disadvantages and advantages. But as you can see from this green plot, there are several non-invasive brain stimulation techniques that we can potentially use for human application. One of these techniques you probably heard about, the repetitive transcranial magnetic stimulation, or transcranial direct current stimulation. As you can see, the spatial resolution of these techniques is relatively big. This is the profile of the simulation. And the temporal resolution is, it can range from seconds to hours. One nice thing about transcranial focused ultrasound, I'm going to call it as TFUS, too short. So TFUS, as you can see, it can deliver a acoustic energy to a very small area of the brain. One of the things I would like to comment on is it can go deep into the brain. So TFUS can offer excellent spatial specificity along with the deep brain penetration. A lot of these things are owed to the development of therapeutic ultrasound techniques. I believe we are in the middle of a Renaissance period for that. For instance, focused ultrasound, it was developed through 80s and 90s, and it offers delivery of acoustic energy to a small, how small? I can just give you an example. It's probably the size of the orzo pasta grain, just give and take. And we can steer the beam into a specific region of the biological tissue. And we can convert or transpose that ultrasound energy to either heat, for example, to ablate uterine fibroid, or you can use it to just deliver pressure shock waves, for instance, to break away kidney stones. For transcranial application, we have skulls in our brain, around the brain. So we have to transmit the energy through the skull. So we have to utilize the low frequency range, which is typically less than 1 megahertz for better skull penetration. For a lot of ultrasound imaging technique that we've seen everyday life, they are typically use about 2 to 3 megahertz, which is much higher than we can use. The reason is if you're using that much of a high frequency, lot of energy, about 100% or 99.9% of the energy will be observed at the skull surface, and it cannot be transmitted to the brain. One of the breakthrough in this technology was seen from the development of this helmet-style multi-array transducer configuration. And there are thousands of elements, transducer elements, that are surrounding the head, and it can be independently actuated and controlled by a computer. And it corrects for all those phase aberrations created by the skull and delivering very focal energy to the brain. But on the other hand, this device is very pricey. And as you can see, it is very bulky. So it is primarily used for neurosurgical applications. For us, a single element transducer, as you can see from this image, I'm going to explain it a little further, can also generate acoustic focus using acoustic lens or the transducer that has a built-in curvature. Due to its simplicity and also portability, you can actually wear it as the headgear type. We are primarily using this configuration. So how do we do the neuromodulation using a transcranial focused ultrasound, TFUS? It is not totally new. It began about 1958 by Professor Fry at the University of Illinois Urbana-Champaign, that they applied the focused ultrasound to the cat, and it suppressed the function, visual processing function of the lateral genic nucleus. And there has been anecdotal studies that has been done in vitro cells and ex vivo brain tissues. But with the help of one of the panelists here, Dr. Bystritsky, back in the mid-2000s, we started to see the in vivo feasibility to demonstrate it is indeed possible. And we kind of did that, and it was followed by many other studies afterwards. But before I getting into a little bit of technical details, I would like to just want to say that the sonication, the ultrasound, is delivered in a pulses. As you can see, small little pulses that has been chopped into small little pieces in time. So the time of on period, we call it pulse duration. You're going to probably see it, so I have to specify, although it sounds a little boring. It's called PD, pulse duration. And we repeat it pretty fast, more than probably 100 hertz, depending on what you want to achieve. And we give it in certain duration in terms of a sonication duration. So we do it because it is not only it give you the efficiency of stimulating the brain tissues, but also you can reduce the energy deposition to avoid the heat generation in the brain. So what would be the potential mechanism behind it? I mean, we've seen a lot of evidences that is the brain stimulation as well as suppression is possible. But we still do not know exact mechanism behind it. Well, but one thing, it is not likely to be temperature related. We just don't deposit any tissue. And number two, it is not really electromagnetic. We don't give any electromagnetic field into the brain. So therefore, most likely it is mechanical mediated. So for instance, ultrasound can induce mechanoreceptor or ion channel activity. And it can also change the cross-membrane capacitance, as you can see from this illustration. One of the new concept behind it is it can also stimulate the astroglial system through a calcium signaling. So just for your note, we actually have more astroglial cells than are the neurons in the brain. So, and recently the scientists started to reveal that astroglial system, which traditionally believed to be a glue that putting the neural cells together, or it could be used as the mediator between the brain cells. It actually does much more than what it does. It actually can communicate. It will also participate in energy metabolism. So there are more things that are being found about the glial system. It can be also mediated by the stimulation of a group or ensembles of inhibitory versus excitatory neurons, resulting into different tissue level responses. So it's not only the individual cell level responses, but it's more of a harmony of the group of neuron cells. And also we have shown that it also modulates the neurotransmitter activity. So one of the things that we found is the pulsing schemes is very important to determine the stimulation versus suppression. With the scope, the limitation of the talk today, I will only concentrate on the stimulatory effects so far. So we saw, through the animal studies that was done in the rats, it looks pretty complicated, but the study shows that you have to kind of use a specific pulse duration at specific duty cycle. Duty cycle, it means how long that you give sonication per given quanta. And we found that about 50% or 70% duty cycle is actually better than low percent of duty cycle in terms of causing excitatory effect of the rat brain. I just want to give you the example. It's not only from our study, the study from Vanderbilt University that is pretty recent. They also get the ex vivo mouse brain slice, and they apply the ultrasound through that, and they actually observed a calcium signal using microscope. And they also found a specific pulse duration actually generate a better neural responses than the one that has been given in short pulses. So all this information encouraged us to try this technology through animal models. We've done about 20 different animal model studies using a rodent. We also conducted about 4 different types of studies on using large animal model, which is sheep. But it is time for us to actually test it if it is safe and it could actually work among healthy humans. So about 7 years ago, back in 2015, we applied the ultrasound to the somatosensor area. And we found it can not only elicit a EEG responses from the ipsilateral side of the brain, but also it can create a different tactile sensation of the hand that are corresponding to the sonication. Then we moved on. In the next year, in the following year, we also stimulated the primary visual area that is under the image guidance. So actually, the healthy individual was located inside of the MRI machine, and we delivered the sonications to the primary visual cortex right there. And then we also acquired functional MRI to examine what kind of activity that we see from the brain that has been accompanied with TIFA stimulation. We found many subjects reported to seeing a phosphene, that is a visual perception. And we saw there's a visual potential, looked like kind of a potential that had been detected in EEG. And we also saw the phos stimulation can yield and activate variety of the neuronal substrate, including the V1 that has been sonicated. So encouraged by that results, we decide to test it out. What if we use the TFAS brain stimulation as a mean to deliver computer to brain interface? So you use the computer to elicit a specific brain response. By the way, we also have a BCI, which is brain-computer interface, that can translate or detect and characterize the brain responses, which can control the computer function. So if you have a BCI and CBI, what you have in the middle is the computer, and the computer is widely connected through the Ethernet. So why don't we create a brain-to-brain interface? So just test that idea, we recruit a healthy participant. They were paired as a sender and a receiver. And the sender performs a motor imagery test to either right or the left hand, and associated changes in EEG mu rhythm from one hemisphere was translated and programmed to move the cursor to right or the left. So now the computer knows what the intention of the sender does. And then this remotely triggers the TFAS brain stimulation. It was transmitted through the internet and can stimulate either left or the right somatosensory area. Then the person can actually feel what other people did. So it was just a brief example of the brain-to-brain computer interface. Very rudimentary, but it's just the idea. So we were encouraged by all this method, but then the question remains, one, what kind of pulse duration should we use to simulate the human brain function? That's one. Number two, we haven't really stimulated the deep cortical areas, for instance, thalamus. That's number two. Number three, we didn't know what's the effect of the TFA stimulation into the brain. Is it just a transient, or can it last a little longer? If you want to have any therapeutic effect, you want to have some remaining effect. Of course, I'm not saying it is negatively affecting the brain function, but it has to last beyond the sonication itself so it can induce the neuroplasticity. And of course, we wanted to evaluate the safety of the TFAS among healthy individuals as well. This study was interestingly funded by NASA because they wanted to find a non-pharmacological alternative to enhance human performance during their deep space exploration. Still, we are far out there, far from exploring the Mars, but they want to gear up for the future exploration. And they wanted to see, in the middle of the lack of supplies, and it's going to be taking a long trip, at least 3 years or so, they wanted to see if there's any non-pharmacological alternative. So the study was approved by the Brigham and Women's Hospital, and FDA, they determined our device was categorized as a non-significant risk category. 8 participants, 3 females, all right-handed, relatively young because they have to be in a similar range of astronauts. 32 individuals applied in the middle of the pandemic. Unfortunately, we were not able to have all of them, and we completed the study from 8 individuals. All those individuals has a really well mix between the genders as well as ethnic distribution. This is the overall protocol. After the consent, the subject underwent CT and MRI. We wanted to have the CT in place because we wanted to see their cranial structures. It was very important to numerically simulate the acoustic propagation through the skull. And the MRI was needed to pinpoint and identify individual primary sensory as well as thalamic sensory motor circuitry. And then about 5 weeks later, we conducted TFAS simulating S1. And then about a week following that, we stimulate the ventral posterior lateral nucleus in thalamus. And about a week later, we conducted EEG. And about 3 weeks later, we finished up with the neurological exam and the resting state MRI. So just want to show you, there has been 3 functional MRI acquisition here before stimulation in visit 1, about 1 hour after stimulating the thalamus, and about 1 month after stimulating the thalamus. I just want to note that the S1 stimulation actually preceded the thalamic stimulation about a week prior. And this is our setup. We had to use 2 different type of configuration. They look pretty similar, but as you can appreciate, for thalamic stimulation, we had to use the little bit larger size transducer. It looked like this. It can reach about 4.8 8.4 centimeter depth, so you can actually go through all the way through across the hemisphere. That's that deep. And for similar, more lighter transducer was to use to stimulate the primary sensory area S1. It looked like this. And although it might be a little bit too complicated, most important thing is we used three different pulse duration, 0.5, 1, and 2 millisecond. And we also included no sonication condition noted as sham condition. And we gave a different acoustic intensity. We show it as ISPPA that are shown here for S1 and thalamus. And we acquired the evoke potential, EEG evoke potential simultaneously. And for abundance of caution, we added a little things called here cavitation detector. It was basically a hydrophone. It's like a microphone that listens if there's any cavitation activity, which is not good, from the brain. But eventually, there is no cavitation event that has been detected, but we had to put it to monitor what's going on. This is brief schematics of where we acquired the EEG from, basically from F and parietal lobe montage, left and right. And before each sonication, we also acquired the somatosensory evoke potential that has been mediated by from median nerve stimulation of non-dominant hand. And this is actually what we got from the electrical stimulation of non-dominant hand. As you can see, the opposite hemisphere shows a bigger dip. Interestingly, in the red one, actually, you can see still a dip, but it was a little smaller than the other contralateral side. So one thing I'd like to note is, well, if you learn from the neuroscience, it's connected, each of the hand activity was related to the other hemisphere, but there's a linkage between each hemisphere. So some of the electric activity may show up in the other hemisphere as well. So this lengthy table, it looks a little boring, but it was very important because, well, we are giving a certain amount of acoustic energy through the skull, but every individual skull thickness is different. Their shape is different. So therefore, we really do not know what exactly the acoustic intensity that are experienced at the in situ focus. Well, it would be great if we have a non-invasive method to image or calibrate the amount of acoustic intensity, but we don't have that technology yet. So the next best thing to do is to run a numerical simulation to estimate where it goes and how much of energy that you are depositing. So long story short, we only have about 37% and 28% of the energy that go through the skull. But it was pretty decent intensity, about 3.4 to 4.1 ISPPA in situ. And the mechanical index was much, much lower than the safety limit. So after we acquired the EEG, for those people who are interested in looking at it, we ran through several series of data selection as well as data filtering to get rid of the power line noise or the 60 hertz related the physiological noises. And then we perform statistical analysis to compare the EEG signature that are coming from different pulse durations. And this colorful image just represent the top is the data that we acquired from the S1 stimulation. The bottom is from the thalamic stimulation. It looks pretty complicated, but it's all color-coded. The EEG features, it's a evoke features are coming from different pulse duration. And one of the things that you can notice is the differences compared to the sham condition actually were more profound only ipsilateral to fuzz for both S1 and thalamic. And among that, use of 0.5 millisecond pulse duration seems like it's a better choice than any other pulse duration that we used. And these results, interestingly, share the similarities that we actually did a similar studies on awake sheep. And strikingly, it actually shared a very commonality. At 0.5 millisecond pulse duration is good, and it's a lateralized effect. So one of the things that we anticipated to see was, well, since we are stimulating the right hemisphere that is corresponding to the right hand, left hand. So we anticipated to see there's probably the difference between EP signatures between left and right hemisphere. But interestingly, we didn't see that from our human subjects. So we believe it is maybe due to the existence of a strong interhemispheric functional connections between somatosensory cortices. And all this work has been done in the non-dominant hand. So there has been a report that if you are stimulating the brain of the non-dominant hand, actually it involves and it recruits the other side of the brain more. So it could probably something to do with that. And also we randomized the stimulation of the pulse sequences, and we probably think it is all these things could be affecting each other. So it may ended up having less than desirable changes that we wanted to see instead of just using one pulse duration condition. So let's move on. So we've seen this EEG signal differences, but what about the subjective sensation that actually the individuals might have felt? So we did a survey, and we found not all of the subject actually felt something. So there's about 3 out of 8 people who felt tingling sensations and tingling movement sensation, or even vibrating sensation from the palmar side. But only one individual felt that kind of thing from thalamic stimulation. So it was a little interesting to see that not all of them felt exactly the same way based on the first stimulation of the brain. One thing that we noted this time was there are majority, not all of them, but 6 out of 8 and 5 out of 8 people, they reported hearing little beep-like tones when they were subject to the brain stimulation. So it was described as a little beep sound. It's faint beep. It's not really loud beep. It's just tangible enough that you can hear it. So we started to worry, well, if it is whatever the evoked potential that we observe, is it because of the brain stimulation, or is it just because of you hear something? So we need to actually conduct a separate measurement of auditory evoked potential by simulating and giving this about the same tone that has been given, or probably they described that it was related to their sonication experience. And we recruited the same number of participants. It was not the same participant who participated in the sonication experiment. It was totally different individuals. And this is the results. So we gave sound stimulation through both ears, and then left ears and right ear. I didn't really include anything from the right ear because it was basically the flat line. So the EEG montage that we use is not really optimized for detecting auditory evoked potential to begin with. If you give the sound signal, it is really audible signals to both ears, and you probably see a little dip that has been related to that. It has a really long latency after the stimulation, which is observed. And let's compare it with the one that we saw before. This is auditory only, and this is from the brain stimulation. So actually you can see there's a huge differences, and then we think that auditory confounders were not likely. So we believe probably the auditory sensation experienced by some of the participants was due to the bone conduction. So when the skull was applied with ultrasound wave that is giving in certain frequencies, that it can vibrate, and then person can perceive a sound through bone conduction. but it was very faint enough so that it doesn't really reflect into our EEG measurement. So let's move on. So the next big question was, well, will the TFUS brain stimulation can induce non-transient neuroplasticity? That was a really big question. So one of the caveat here is functional changes that last greater than about 35 minutes or more are known to alter synaptic changes that may induce neuroplasticity. So 30-minute time window is the one that you really want to target on. So to examine it, we decided to use a functional connectivity analysis. So what it is the functional connectivity analysis. I'm going to give you some of the example later on, but it may show the long-term effect of TFUS, such as neural plasticity. So what it is when you acquire the resting state fMRI time series data, just while the subject is not doing anything, you just close their eyes and they should not sleep, but they should be awake but yet rest in the middle of the scanner. Then we run through and then we kind of acquire the temporal signal from each of the pixels in the brain, and then we run the correlation if there's any correlation between the blood oxygenation level changes, bold signals, and it could have a positive correlation, but it could have an opposite correlation like this. But if it has nothing to do with any hemodynamic changes between the brain, there would be no correlation whatsoever. It would be just like randomly mixed together. So we thought that I'm going to go back to our protocol here. We acquired the MRI, resting state fMRI 3 times before for TFUS stimulation, about 1 hour after first stimulation, and about 1 month after the thalamic stimulation. And then we compare the data between 3 different sessions. And again, this is a bit of details that to those people who might be interested in looking at it. And we had to put a seed region of interest. You have to kind of start from somewhere because you have to kind of set the reference point where you find your relation, functional connectivity relation into something. So we put a seeding area in the sonicated somatosensory area. That's one seed area. The other seed area was sonicated right thalamus that has been described here. And then we run a functional connectivity analysis, and the acquired correlation coefficient was converted to z-score. And then we process the group analysis. So this is example from one individual. It looks very colorful. So it just shows you from one person with respect to the S1, that how of these small little brain, the image pixels are related to the time signature of the S1. So the red indicates positively correlated. The blue is still correlated, but it's negatively correlated. The yellow and green, it means it is probably not correlated to each other. So the same thing actually can be conducted for the thalamic area as well. So this is the grand sum results that has been gone through the group level analysis. So this area shows representation of the group level, by the way. So when you compare the visit 3 and 1, which is, so this entire slide is to detect the functional connectivity changes 1 hour after TFUS. So we saw the enhancement in the resting state functional connectivity in Brodmann area 40 and 42, which is sensory association area right here, that is ipsilateral to sonication, as well as sensory motor areas. We didn't stimulate the motor area, but somehow the motor area has been involved and enhanced contralateral to the sonication here. In terms of sonicating the thalamic area, we saw there are a variety of insula or putamen, which is subcortical areas that are involved in a lot of different somatosensory processing, and high-level sensory integration areas such as Brodmann area 4622, right here, ipsilateral to sonication, and Brodmann area 102145. It's a lot of veterinary area here that has been that are contralateral to sonication. So actually, you can see there's a vast amount of enhancement just appear 1 hour after applying the TFUS. What happens? What kind of things happen after 1 month? It quiet down quite a lot. There's this area that has been initially elevated, now is went back to the baseline. And some of the insula putamen area activation, as well as Brodmann area in middle cingulate circus and pre-central gyrus, that activity has been went down at 1 month afterwards. So when we directly compare between the very first session to the one that has been acquired at 1 month later, we didn't really see any difference. It means that whatever the changes, the functional connectivity changes that has been appeared by the sonication has been restored. Well, one thing is that, but there's usually 1 hour, we definitely know the 1 hour. The effect of thalamic stimulation lasted more than an hour, which is 82.5 minutes in our case. But what if the stimulation, the effect of stimulation can last more than 1 week? Remember that we conducted S1 stimulation about one week prior to that. So it might have mixed together. It still appear as the time that we acquired the second resting state fMRI data. So if we are given a chance, we really want to conduct a more longitudinal studies that you can actually isolate the effect from either S1 or the thalamic area stimulation. So the lesson that we learn is the effect of TFUS may outlast the stimulation session itself, definitely, at least about an hour or more. And as I mentioned before, about an hour, maybe it is sufficient to induce the neuroplasticity in the brain. And therefore, it could be used for a lot of different therapeutic applications. To wrap it up, we conducted a multitude of neurological exams. We appreciate our neurologists for their dedications to allow us to do this the neurological exam. Everything turns out great. We also measured the brief EEG. It was normal across all individuals. And we conducted 3 different MRI sessions, anatomically reviewed by 8 different radiologists who were blinded to the study. And there was no abnormality detected across the participants. So here is my concluding slide. Specific sonication parameters exist, especially regarding the use of PD pulse duration, to yield a superior stimulation efficiency among humans. And it kind of shared by different species as well. And the sonication of TFUS outlasts the stimulation itself about an hour or more, and it may be sufficient to induce neuroplasticity in the brain. So far, the TFUS has been safe. And of course, we wanted to conduct further studies on suppressing the original brain function in humans. That would be great. These are the funding sources to make this project possible. And that's about my presentation. Thank you very much.
Okay, I told you that was going to be good. That was amazing. Thank you so much, Seung-Schik. We just have about 5 minutes for questions now. If anyone has a burning question they want to ask, or we could also save them to the end.
Hi, can you hear me?
Yes.
Thank you so much. That was a great presentation. I did have a couple questions about the protocol and just balancing out controls. So it looks like when you were looking across the different pulse durations, your experiment also modulated the pulse repetition frequency, I'm assuming, to balance out the temporal average of the stimulation. Were you guys able to do any kind of pilot testing or determination that the differences you saw in the stimulation were not just related to the differences in the frequency of the stimulation, but actually due to the pulse duration itself?
That is really, truly an excellent question. So I just want to give you a little further additional explanations to that. So the reason that we changed the pulse repetition frequency is to fix the duty cycle. So we fixed the duty cycle into 70% or 50%. That's the whole reason behind it. Yes, it will be interesting to see. Now you can actually fix the pulse duration and use different pulse repetition frequency. But now another thing is that you're probably going to change the duty cycle along with it. So that will be interesting questions to ask. But according to our experience with the small animal studies, I think the duty cycle is more of a factor. But also there's in vitro studies from other groups that are indicating the choice of pulse repetition is also important. So the jury is still out there, but obviously it requires further investigation. I think it is both factor of a pulse duration, duty cycle, and pulse repetition frequency. So it really gonna depends on which one you fix, or you study all the potential combination somehow and determine it.
Okay, cool. Thank you.
Thank you very much.
So with those MRI changes that you guys observed, were you able to determine that that wasn't just from the effect of going through a somatosensory evoked potential protocol and, I guess comparing the active TFUS stimulation compared to the control? Because I just was wondering about that, because I do have experience with somatosensory evoked potentials, and at the amplitude that you're using, you do get this reliable response, but in an awake person, there's also, it can be painful. It's very noticeable sensation. So I was just wondering if you guys kind of determined that those changes were not just because somebody went through somatosensory evoked potentials and not due to the ultrasound.
That is an excellent point. We didn't really account for that effect because, well, perhaps we should have, but retrospectively, our SSEP protocol was very naive. It was very short. It was basically done in under 2 minutes.
Okay.
We cannot completely rule out the fact that there's nothing to do with our observation, but I think that it probably didn't affect. One, the stimulation was very, how should I say, favorable stimulation. It was not painful or anything like that. And number two, it was really short. So probably we can do those experiments alone, but I think it would be very interesting. Just this like 2-minute stimulation of median nerve can actually have a long-lasting effect in the resting state functional MRI. That will be also interesting.
Mm-hmm.
Thanks for the comment, though.
Yeah, thank you so much.
Thank you for those questions. I think we can have a very, very quick one.
So there has been the question that has been appeared in my chat lab, chat window. Mm-hmm. Can I read it for you?
Sure, go ahead.
So why did you perform both CT and MRI? Wasn't MRI alone enough to have all spatial informations? I think it is a really good question. So actually, it's not, the MRI is not really optimized to detect the bone structure. So the reason that we conducted CT is one, to get the cranial structures, the CT is still the best. People are still researching on how do you use MRI to detect the skull structures. For instance, ultra short, the time of echo MRI sequence is one that has been suggested, and it is heavily researched field. But we are not really there yet. Still, the gold standard to get the cranial structure information is from the CT. Hopefully, I answered that question.
Yes, thank you. Go ahead.
Okay. Well, thank you for this presentation. This was great. When you were looking at the ERPs that were concurrent with the ultrasound, your stimulation period, you were stimulating people for like 20 minutes.
Right.
Right? You're doing this for a long time. Did you notice, or did you look to see if there was any accumulation effect of the way the EEG looked early on when you first started stimulating to doing it across time? Was there any kind of change in the accumulation of energy there?
Right. That is also excellent. I'm getting really good feedback today. Thank you. So we took a look at that features. We didn't find any. One of the things I like to describe a little further is all this quanta of simulation was delivered every 5 seconds, and it was computer generated randomized fashion. So we do not even know what the subjects are experiencing in terms of the pulsing sequence. So we didn't really see that much of a time-dependent features in ERPs.
Okay, thank you.
Thank you.
Oh, thank you for the great questions and the amazing presentation, Seung-Schik. Thank you so much. So we'll move on to our next speaker, Dr. Martin Monti, who is calling in, I believe, from UCLA, but from the background, we can't tell where you are, so maybe you're in space or something. So thank you for joining us. Yes, thank you for joining us digitally. I have a long intro, but I'll save it. There's lots to say about Dr. Monti as well. He's a professor at UCLA, done so much interesting work, particularly for the field of consciousness studies. Just one study that I love from him, where they attempted to communicate with people who are locked in. So these are people who have had some brain trauma, and it's not clear how conscious they are, from vegetative to minimally to fully conscious. And so he's designed these super clever paradigms where you use something like an fMRI to attempt to try to communicate with these people to see how conscious they are, whether they can respond or not. And he's sort of applied that clever methodology now with ultrasound to attempt to elevate the level of consciousness in people who have had brain trauma. So I think he's going to talk about that and probably some other interesting things today. Martin, I learned also from the internet that you're a pilot. So the next time I visit you in LA, I hope that you can take me up and show me Los Angeles from the sky.
That would be a great pleasure, Jay.
Yes. All right. Well, thank you.
All right. Well, thank you so much for having me. This is just wonderful. It's just great to hear Seung-Schik sort of go through all of the evidence, both sort of the less recent and the very recent one. I learn every time I participate in these type of meetings. So here's what I'd like to do today. You've heard from Seung-Schik the science and the really interesting things that can be done using ultrasound as a neuromodulatory technique. I would like to show you one type of application that we've been leading over the last, 8 years, 9 years now, which is the application of low intensity focused ultrasound to a specific clinical population. I guess I should probably start by just saying that I don't really have any disclosures, but I do have some very generous funding that allows us to do the work that we do. So here's the plan for what I want to share with you. Just a little bit of background about the particular clinical condition I'm interested in, which is called the disorders of consciousness. The tiniest background on low intensity focused ultrasound, which is exactly the same that Seung-Schik calls TFUS, and there are other 15 different names for it. Hopefully, one major achievement of the field will be to converge on one acronym as to what's the name. And then I want to show results from a very first first-in-man feasibility proof of concept trial, and then some early results from an ongoing trial, trying to use ultrasound to help patients recover. So let me start from this question. What are disorders of consciousness, in case you're not familiar with this, if this is not something you have encountered before? So disorders of consciousness is a set of conditions that describe patients who typically survive severe brain injury, traumatic or non-traumatic, but they don't quite fully recover a state of consciousness. And of course, the reason behind this is that at the core of my interest is how does consciousness emerge in the human brain? And so this is one way of addressing that question. Now, you might be familiar with the state of coma. This is defined as a state of unarousable unresponsiveness. So from the clinical point of view, at least using some of our clinical reductionism, we look at these patients, they don't show any sign of arousal in the sense they look as if they were permanently asleep. And they don't show any sign of being conscious of themselves or what is around them. They will respond to the environment if you stimulate them, if you stimulate reflex pathways, but they will not show any purposeful response to the environment. Now, many patients from coma just recover, and some patients, however, a sliver of patients, enter other disorders of consciousness. What is known as the vegetative state. Now, this is a condition where, very paradoxically, you will see patients open their eyes. They will open their eyes and close their eyes as if they were waking up and falling asleep. Whether they truly fall asleep in the way that a healthy individual does is not necessarily clear. They will wake up and fall asleep, so their eyes will open. They will sometimes move and exhibit spontaneous behavior, but they will show no sign of being aware of themselves or their circumstance. So these patients are awake, but not aware, and they don't seem to have any self-awareness. And that's what makes it so difficult. A patient with open eyes, that normally when somebody has open eyes and they are looking, they are seeing, they're having an experience with that looking. These patients don't seem to have, or at least don't give any indication that they have any type of experience. Now, some patients remain in this condition for the rest of their biological life. Some patients recover the ability to demonstrate signs of consciousness, and they enter what is known as a minimally conscious state. So this means that, again, they have some level of wakefulness. They wake up and fall asleep. So they have these rhythms. I'm so sorry. Give me a second. I'm sorry. They give signs of, so they show diurnal rhythms and arousal, but and they can show small signs of being conscious. These could vary from being able to move a foot on command, move a finger on command, blink on command, these kind of things, or simpler behaviors like tracking an object in a room, these kind of behaviors. In fact, let me give you an example. And Jay, this is interactive. I'm going to need your help. If you could look at the screen here, you will see the clinician who's doing what is known as the Coma Recovery Scale Protocol on this patient. And you can see that sort of she's doing what is known as a startle response. And the patient closed her eyes every time. Now, if I were there, I would look at the crowd and say, how many of you think that this patient is conscious now that you've seen this behavior? Is there any chance I can get a little feedback of how many hands come up if you see this? Is this person conscious? All right, you'll have to make a mental note. Maybe when we're doing questions, I'll ask for what was the gestalt in the room. How about this one? All right, so that was a simple sort of response, but here's a second one. You ask the patient, please follow this object with your eyes. And now you, the clinician, have to decide if this patient is conscious or not. Okay, this is what it boils down to. Now look at her eyes very carefully. Okay, question number 2. This is the same patient as before. Is she conscious? You're the clinician. It depends on you. Her diagnosis depends on you. Is she conscious? I would ask you to raise your hands to say yes or no. If you want to do it, Jay, give a quick scan in the room, and then later I'd like to hear what the response is.
I'd say about 10% of the audience says she's conscious.
How about the first one?
About 20%.
Okay, very good. Thank you. Thank you very much. Turns out the first one is not enough to say that this person is conscious. It's a reflex. It's not mediated by consciousness. It needs 3 neurons. So it doesn't meet the standard to be a demonstration that the patient is conscious. The second one, if she had tracked that object, it would be enough to say that the patient is conscious, but she didn't show any sign of it. At this time, this patient was considered in a vegetative state. And also one important aspect, everything I'll show you from now on, the dependent variable is how much a patient responds in these kind of tests, known as the Coma recovery scale revised. It's just a clinical test that we do to assess the depth of the impairment of consciousness. Okay, so this is what we're talking about. Now, why would we want to use ultrasound in these patients? Well, simply put, if patients don't recover spontaneously, there's really very little we can do. There's just no real interventions available for these patients. In acute patients, there is some, there is the best evidence today is that you can give amantadine and it might speed up patient recovery, not change it, but speed it up a little bit. But beyond that, there's really almost nothing. And so the question is, can we do better? Can we help these patients recover some level of consciousness after severe brain injury? Now, of course, in order to stimulate consciousness, you need to have some understanding of what does it look like in the brain? Why do these patients lose consciousness? It turns out just about 20 plus years now of research argue for the idea that disorders of consciousness are a dysfunction of the thalamocortical circuit, which you can see sort of pictured, depicted here with sort of cortical sides, basal ganglia, striatum, lobus pallidus, and thalamus. And it really seems to be a dysfunction within this circuit that correlates with the depth of the impairment of consciousness. In fact, if you look at patients, the one clear characteristic of these patients is that the more impaired consciousness is, the greater the metabolic depression in the brain. So this image down here, if you're not familiar with it, is a positron emission tomography image showing the amount of metabolism that is going on at various cortical sites. The redder, the hotter, the greater the metabolism. And you can see a clear gradient with healthy volunteers, so you and me. And then patients who have survived severe brain injury have some level of consciousness, so a minimally conscious state. Patients, the same patients who have survived the severe brain injury but did not recover consciousness. And then patients in a coma. And you might notice that the deep central parts of the brain seem to be particularly disproportionately affected and hypoactive. And indeed, while there's no doubt that consciousness is a network affair in the brain, there are some parts of the brain that seem to have a privileged connection to allowing the emergence of consciousness, and in particular, the thalamus right in the middle. It's well known that a lesion to thalamus will send someone in a coma immediately. Patients who die in a vegetative state will often show, I know it's hard to see, but you might see a little black spot right here and a black spot right there. Patients who die in a vegetative state will show either great atrophy of the thalamus or lesions in thalamus. If you do the same study in vivo, and you look at, these are the basal ganglia and thalamus, seen from above, left and right. The red areas show areas that are atrophic in these patients in proportion to how deep the impairment of consciousness is. So clearly there's a relationship between the damage or the atrophy in thalamus and the impairment of consciousness. Not only, it's not just thalamus itself. If you track the cables, the white matter that go from thalamus to frontal and prefrontal cortex, for example, you will see See, the colors down here tell you how many tracks we can reconstruct from thalamus to cortex. Patients who have a lesser impairment are in a disorder of consciousness, but with lesser impairment have a lot of intact tracks that we can reconstruct using diffusion tensor imaging. The deeper the impairment, the fewer the tracks we can reconstruct from thalamus out. So not only thalamus is getting either damaged or atrophic, but the cables that connect thalamus to cortex are thinning out, okay, in proportion to how deep the impairment of consciousness is. Based on these ideas, we've developed a number of experimental interventions to help these patients. To give you sort of the most studied examples, people sometimes use transcranial direct current stimulation, which is a non-invasive technique. The idea is to stimulate cortex, and through its secondary innervations to thalamus, if you upregulate cortex, you're sort of trying to upregulate thalamic activity and try to modulate thalamic activity indirectly, which is great because it takes no surgery, but it's an indirect approach. Alternatively, people have administered amantadine, which is a dopamine agonist. And amantadine seems to act at the striatum. Its net result is that it seems to allow thalamic output to reemerge and metabolism to reemerge. Very paradoxically, if you give zolpidem, which you might know as Ambien, which is a sleeping drug, a hypnotic, actually, it seems to help patients, a small sliver of patients, just about 5%, after they're administered after they're administered zolpidem or Ambien, recover consciousness. And then they lose it as soon as it washes out. And this seems to be through action to the globus pallidus. I'm sparing you some of the weedy details, but if somebody's interested, I'll gladly go over them at the end. And finally, the one to me of most interest, and of most interest for today's purposes, deep brain stimulation. So you can actually put leads inside the thalamus, and you can see here the tracks left by the leads, and inject energy directly in thalamus, and this will actually help patients do better. So I really want to focus on this one. And in fact, not only we have seen this clinically, two recent studies that were published last year and this year showed that you can awaken a non-human primate from anesthesia, from a steady anesthesia with deep brain stimulation to thalamus. Here's one example. You can see the eyes of the animal during the anesthesia. Then suddenly they turn on the stimulation, the eyes open. And then once you stop the stimulation, the eyes close again. The animal sort of appears to fade back into unconsciousness. And if you look at their behavioral response to an air puff to the eye, you can see how suddenly during the stimulation, they start responding a lot more so than baseline. And although this is a slightly more crowded image, a second group has shown exactly the same idea. I will really focus you on the top right corner of this graph. This is their the degree of arousal before, it's a composite score, but the degree of arousal before stimulation, during stimulation, suddenly they seem to be more aroused, and then after the stimulation, to thalamus, again, with a deep brain stimulation. And you can, if your eye is used to EEG, you might notice that there are some very big, powerful deflections during the periods of unconsciousness. Suddenly you turn on DBS and you get desynchronization, which is more typical of wake states. And then once you stop that, slow rhythms that are typical of unconsciousness reemerge. So clearly now there's converging evidence that stimulating thalamus seems to be able to alter some basic aspect of consciousness. And so, of course, to us, the question was, can we do this, right? How well can we do this type of intervention? Unfortunately, there was a prospective 5-year clinical trial, actually 7-year clinical trial, where they surveyed 40 patients in a disorder of consciousness. They were trying to evaluate them to see how many were eligible for deep brain stimulation. Of these, 5 were selected to undergo deep brain stimulation. Only 3 could actually receive the deep brain stimulation. And of these 3, ameliorations were shown. So they did show ameliorations in their clinical score on the test that you saw earlier. So this confirms the benefit of deep brain stimulation. The problem is that it also shows that there's just about an 87% of patients who had to be excluded from this clinical trial. So it's efficacious, but it's not viable. And so to us, the real question was, can we do the same without the surgery? Can I actually use a non-invasive technique like focused ultrasound, just put a transducer by the side of a patient's head and directly stimulate thalamus, non-invasive? So this is the promise to me and to my field of low intensity focused ultrasound, of the work that you've heard from Seung-Schik. And that you sort of hear, and the further work you'll hear in the rest of the session. And indeed, there's now good evidence. You've already heard much of it, so I will only show 2 data points, that focused ultrasound is bioactive. In fact, is neuromodulatory, particularly in deep parts of the brain. You've heard some evidence in the previous talk. This is just a recently published paper by our group looking at changes in bold response, so functional MRI, during stimulation to the globus pallidus, so deep brain region. And what I want to highlight is you can see here in blue the decrease in the bold signal, the significant decrease in the bold signal in the globus pallidus, which was our target area, as well as the thalamus, which is the mechanistically downstream part of the brain. And if you look sort of at a full brain level, this stimulation does lead to full brain changes in resting state. One small side note, somebody asked about different parameters, different pulse width, and different repetition frequency. The graph on the left is showing our conventional approach, which is 100 hertz pulse repetition frequency, 0.5 milliseconds pulse width, and a 5% duty cycle. And it has an effect we can demonstrate. If you actually stimulate still with a 5% duty cycle, but with a 10% with a 5 milliseconds pulse width and 10 hertz pulse repetition frequency, we don't seem to get an effect that we can detect with functional MRI, and that are the red box and whisker plots that you're seeing right here. And we don't see anything at the full brain. But for our purposes, everything that I'll discuss today will use the parameters that I just mentioned that are shown here in blue that have been shown to be bioactive. And the only other piece of evidence I want to show is from Dr. Yu's group, because it's directly relevant to what we do. So they used a rodent model of animals who were anesthetized. This was an intraperitoneal xylazine ketamine anesthesia. And at the 40 minutes mark, rodents either underwent sham ultrasound or ultrasound to thalamus. And then they measured how quickly both reflexive and non-reflexive behaviors returned over time. And what you're seeing here is, in a sense, that this graph is laid out in complexity with less complex and reflexes on the left and sort of more complex motor behavior to the right. And so if you look at things such as the increase in respiratory rate, rapid and irregular respiration, movement of the whiskers, and eye blink response to puff, there might be some numerical differences. What is so really interesting is that the animals who underwent real sonication that you see in gray, they recovered faster response to hin-paw pinch and the return of full voluntary movement. So this is yet another data point that shows that you can manipulate aspects of consciousness, in this case, the return of consciousness after anesthesia, by modulating thalamus, not only with DBS, as you saw in the non-human primate model, but also with ultrasound. So on the shoulders of this work, we thought, let's try to see if we can have a beneficial effect in patients in a disorder of consciousness. So a few years ago, we initiated what was a feasibility clinical trial, really a first in man. I think at this point, this was the first example of applying ultrasound to patients. And the idea for the trial was fairly simple. We assess patients on the day before and on the day of the ultrasound with our clinical battery, which is known, as I said earlier, as a Coma recovery scale revised. Then they undergo MR-guided focused ultrasound, which means we put them inside the MRI. And we aim at thalamus with the MRI. And you can see on the right side an example of our transducer, which you can see here on the side of the skull, which is aimed in a straight line to thalamus. This is not a numerical stimulation. This is a point and shoot type approach. And sort of this is the same image with a different contrast because it shows nicely the thalamus right here. And just to give you an example, when we try to reconstruct where we think our focus happened, our focus was, you can see here three sample patients. In the yellow, we're highlighting thalamus. And the two dots are the 2 points of where we think our ultrasound was aimed. So essentially, we're just confirming that it looks like we were indeed reaching thalamus in our patient. In terms of what we think we are doing energy-wise, let me give you actually, we ran some numerical stimulation through a CT skull. And what you can see here is a coronal cut of a skull. And in green, you can see the parts of the, you can see tracks where the energy that is deposited by the ultrasound is above 600 kilopascals. And I want to focus your attention on this little cigar of energy right here. That's what we're doing. In fact, let me zoom into it and make it a little sort of easier to see on the eye. If this is sort of the ultrasound that is just by the skull, you can see a lot of the energy gets deposited and refracted at the skull. And then there's a focus of energy at the time, just about a centimeter and a quarter long. And that's where we think we are being, the bulk of the bioactivity is being deposited. If you want a visual for it, this thing that you're looking at is our old transducer, our BrainSonics transducer. It's inside a bucket of water, and it's pointed upwards. It's pointed at you. When you turn it on, here you can see the surface of the water. When you turn it on, you can see the vibrations right here. So that's what we're doing. We're literally vibrating neurons. Okay, and you can see its effect at the top layer of the water right here. So that's what we're doing. So how has it been working out so far? Here are the clinical results from the first in human study. One small premise. This, because it was a feasibility test, we did in acute patients in the ICU. So these are patients who are in a coma, in a minimally conscious state, in a vegetative state, but it's early on. It's within the first few months from the brain injury. So typically, all of this happened in the ICU at UCLA. And then we also had a sample of chronic patients. So these are patients who have been in a disorder of consciousness for at least a year. And what I'm going to picture is on the left, the greatest score they ever achieved before ultrasound on the clinical test, with higher scores being better. And on the right side, on post, I will plot the scores after ultrasound. So this is our acute population. And this is what happens when we look at their best behavior after. And there's a significant increase in behavioral responsiveness. Typically, we just see patients typically doing more responses than we see before ultrasound. Now, it's not earth-shattering that patients after a couple months from a brain injury recover some aspect of responsiveness and consciousness. It happens all the time. Of course, we enrolled patients in our study because they were not recovering. That's the reason we enrolled them. But patients recover spontaneously all the time. So this is a feasibility study. It's hard to take this as hard evidence that our treatment in particular is doing the job without having a double-blind setting. What is much more interesting is when you look at chronic patients, these are patients that, as I said, have been in this condition for over a year, which means they've met the threshold for chronicity. Although they do show some recovery, usually it's characterized over the span of months and years, not a couple weeks, which is the length of our intervention. And in these patients, so here you can see the scores before. I should say, on the left, you have 11 patients, and on the right, you have 10 patients, which is as many as we could acquire before COVID shut us down. And here's their best behavior on the Coma recovery scale at the beginning of the procedure, and this is their best behavior, their base score, clinical score after the ultrasound intervention. And so again, there's a very clear significant effect. And here, we don't expect it just by chance. These patients just don't change this fast. It's just not, there's been a lot of studies characterizing how these patients change over time once they have met the threshold for chronicity. They do ameliorate, just not that quick, not in the span of a week. So this is much more intriguing evidence in favor of ultrasound having some effect. I should also say that the baseline and the post, these are collected typically, that the maximum distance is typically about a week. So that's the maximum time interval that we've looked at in terms of whether the effects are permanent or sort of how long they might last, rather. A second interesting data point I want to show for this particular study, focusing on the acute cohort only, is that we looked at their fMRI, at their bold imaging, during the ultrasound. Now, first, if you look at the periods during which we stimulate the thalamus, you do see some effects, some brain-wide effects, and you can see them here marked in blue, which also means that what we're seeing is that during thalamic stimulation, these particular parts of the brain, their bold signal decreases compared to when the stimulation is off. So in a sense, we have it's incorrect to interpret it as a depression effect, because that's just not how the bold signal works. But certainly the bold response is lower during ultrasound than it is during the periods of rest. Not only, if you take thalamus and you look at what parts of the brain co-vary with thalamus, so essentially what's the network strength of thalamus, during the ultrasound, there was greater connectivity between thalamus and some cortical sites in motor and prefrontal cortex. So not only we're changing thalamus itself and some remote areas, but actually we're changing the connectivity of thalamus during the stimulation. What is very interesting is that if you look at the thalamus we did not ultrasound, so the thalamus on the opposite side, it didn't show any change in its functional connectivity. So we're changing one thalamus, the one we're targeting, but not the other. Even more interesting, and this is where I think this is an important data point, while, as I said, we have to be cautious in interpreting any causality between ultrasound and behavioral changes, because we don't have two conditions, a sham and a control. If you look at the relationship between changes in thalamic connectivity, all the changes that you see here are correlated to how much the patient will ameliorate a week from now. So the changes that you see here are the changes in thalamocortical connectivity that predict and are associated with how much a patient will show improvement in the clinical scale. So in a sense, this is linking, there's some kind of physiological link between what we are doing at ultrasound, the changes we're inducing during the ultrasound, and the behavioral changes we're going to see over the next week in these patients. Again, interestingly, if you do the exact same exercise with the other thalamus, the one we did not sonicate, we don't see anything. There's no association between that thalamus, its connectivity, and behavioral ameliorations later on. So this was, very enthralling initial evidence. And again, I try to be very cautious because nobody's blinded so far. This was just a feasibility type thing. But then we took this, and during COVID, we thought, okay, let's do something a little bit better. And so we're now doing a new clinical trial, and I just want to show you a couple data points of early results, where we bring patients to our facility at Casa Colina in Pomona, just about an hour away from UCLA. And we first keep them for 4 days, and we do a lot of baseline measurements, which include behavioral clinical measures. Then we do electro-EEG, a couple baseline data points. We do PET, positron emission tomography, and we take an MRI for aiming purposes. We administer at the bedside, so no longer inside the MRI, but at the bedside, with a setup similar to the one you've seen in the previous talk, and I'll show you an image. And then after, we do a number of follow-ups over the next 6 days, including, again, clinical, so behavioral responsiveness, more EEG, and more PET. The setup looks somewhat like this. So here you can see my team. The patient is here, and sort of the face is blurred, so that's the black that you see. But the transducer is on the left side of the patient's head, just by the temporal bone thinning, which is the best point for us to access thalamus, because it's the thinnest part of the skull. And instead of doing this, instead of aiming through the MRI, we are using a stereotactic device, a frameless stereotactic device, which you can see right here. So this device is seeing both where the head is, where the transducer is, and is helping us aim. Indeed, you can see that both members of our team are staring down this little screen down here, which is showing how well we're targeting our target in the brain. So I know it's hard to see, but there's a little tiny spherical marker in the middle of the brain that is marking where thalamus is, where we want to intervene. And we're just using this to make sure that we are on target. So this is the new setup, which is much better because we can do this at the bedside. We don't need an MRI to do the ultrasound. In a sense, it's very similar to the setup you've seen in the previous talk. Now, looking at the same setup, portrayed here are 5 patients, but I can already tell you that there's 8, and 7 out of 8 have the exact same pattern. Before the ultrasound, after the ultrasound, in terms of their amount of responsiveness. It's already significant at 8 points, but it's a small data set for now. So the behavior is still there, and we still see these increases in behavior. As I said, they typically manifest as more responsiveness and new types of behavior, like being able to more systematically move a finger or open their mouth or blink or something of that sort. The last patient actually showed the ability to start vocalizing in response to questions without sort of quite articulating words, but they would vocalize, which they didn't quite do before. So this is the clinical data. An interesting data point, the EEG data. And although we acquired multiple times, I'm just showing you the EEG just before and just after. So the difference between baseline and post, just about 35 minutes at most. What you're seeing here is a relative delta power, how strong the delta power is. See, in these patients, delta power varies. The strength of delta power varies with states of lower consciousness, with greater impairment. It's not a perfect indicator, but patients who are unconscious or more impaired tend to have stronger delta power. And so if you look at the relative delta power, it starts up here, and then right after ultrasound, it comes down. And if you look at beta power, you see, so a faster frequency, you see exactly the opposite pattern. We don't quite know if this maps onto greater information transfer or greater information flow. We haven't done more complex analysis yet. We're reserving those for once we have the full sample. But certainly we are seeing in the span of 30 minutes a measurable objective change in biomarkers. clinical assessments are the standard, but they're also subjective in a sense. This is objective. There's nothing that we can't mess with this. So it's very intriguing. And then a last point for anybody who wants to use ultrasound, because we just found this to be important in our data, and they might be important in yours too. So a paper very recently, at the beginning of the year, I believe, came out showing that there's a relationship between how the brain is before you do ultrasound and how effective ultrasound is. So this was done in a rodent model where they could look at local field potentials. And essentially, the slower the EEG was, the less effective ultrasound was. The faster, the greater the preponderance of fast rhythms. In this case, they looked at, for example, gamma, delta. Then the greater the preponderance of faster rhythms, the more effective ultrasound was in terms of local field potentials. And so we thought, well, this would be amazing for us to have a biomarker of susceptibility, to be able to know beforehand who will respond and who will not, or who has a greater chance of responding and who doesn't. And so we looked I believe these are 5 of our patients. This is what we see. So a pretty convincing, we've started doing this also with 2 of our newest patients, and the trend is exactly the same. We do see a very clear, significant association between how fast, between the relationship between fast and slow rhythms just before ultrasound and how effective the ultrasound is. So if at baseline, if just before the ultrasound, the patient is exhibiting faster frequencies, that will lead to greater behavioral change after. So it will lead to greater efficacy over the next week, not just right after, over the next week. Again, we are predicting with the ultrasound, with the EEG just before ultrasound, how much a patient will ameliorate over the next week, not right after, over the next week. So this is starting to give some link between what we are doing with the ultrasound and the amelioration after. And with this, let me just sort of bring a couple points together. Overall, focused ultrasound, transcranial focused ultrasound, definitely a very promising technique, I would say, for both brain mapping and applications such as those you've heard in the previous talk, and clinical interventions. In a sense, to me, it's a bit like the best of both worlds. You can directly target deep parts of the brain, for me, the thalamus, but in your case, you might be interested in the amygdala, the basal ganglia, ventral striatum, all those parts of the brain that are very difficult to access without surgery. So it can directly target thalamus like deep brain stimulation, unlike transcranial direct current stimulation. But of course, it's non-invasive. So we can finally, for the first time, modulate deep parts of the brain without requiring surgery. We've never been able to do this before. Right? Ultrasound is what is giving us this possibility. In terms of safety, we've never seen any change in vital signs. All our patients are on monitor throughout the whole procedure, obviously. We've never seen any change, any physiological change in terms of heartbeat and CO2, things like that. No change in vital signs. We have seen some adverse events. For example, one patient had a seizure 1 week after, but the medical team that reviewed it thought that it had nothing to do with our intervention. It was due to sepsis that the patient entered. One patient had a respiratory depression before ultrasound, so on their way from their bed to the MRI machine, so definitely not related to us. We had a couple things, an upper respiratory infection in a chronic patient, a fever due to an infection to an elbow, to the cubitus, in fact, to an elbow. So obviously, these patients have adverse events. It happens all the time. None of these seems to be causally linked. There's no obvious mechanism to causally link what we've been seeing to ultrasound itself. They might be linked to the procedure, like putting them putting them in an ambulance, bringing them to us, et cetera, et cetera. But so far, we confirmed the safety record of ultrasound. In terms of effectiveness data, I think I cautiously say potential synergistic effects in acute patients. But I do think we're starting to have preliminary therapeutic effects, evidence of therapeutic effects in chronic patients. But I'll remind you, this is all done open label. We're looking forward and trying to gear up for a randomized sham controlled clinical trial. Last, a lot of questions remain unanswered. What are the exact mechanisms of why is this helping in focused ultrasound in the DOC patients? We're not really sure. We hope to gain some information from the clinical trial we're doing right now. What are the best parameters? You've heard how important parameters are, both in this talk and the previous one. And of course, just about how long these effects last after the week that we see them, we don't really know. And this, of course, will be crucial to know if we need to do multiple interventions or things of that sort. And with this, let me just say thank you very much for your attention, and thank you to all the people that have contributed to this work.
All right. Thank you so much, Martin. Amazing work. Really inspiring. I think we have questions, time for about one question, and then we'll take a short break. Any burning questions from the audience?
I have a burning one from someone who's online who asked a really good question. Can we do this with patients when they underwent craniectomy? Some patients, of course, because of what happened to them, they replace sometimes part of the skull with other type of materials. And the answer is that the one time when we had a patient like that, we decided to stimulate the other thalamus. I was just not confident about what are the properties of absorption and refraction of craniectomies. It's a study that begs to be done, but it's just not my lab, the proper place to do that. So far, we've just used the other entry point on the other side, but it's definitely something important. And we do need to model, we are now modeling each individual patient to see what we're doing. Again, you heard some of the importance of this in the previous talk. And if you don't mind, there's a second question that I'll quickly answer, which were our EEG biomarkers were calculated across the whole brain. They were not site-specific. So they're averaged across multiple channels in a standard 64 montage.
Great. Thank you. Any other questions? All right. We're going to take a 5-minute break just so you can stretch your legs. Quick bathroom break. And we'll start with Dr. Sanguinetti next. So I'm next. Okay, welcome back. Good to go? All right. Well, thanks for sticking around. So we have 2 more talks up for today, and then we'll have a panel discussion after this if there's time. So I'm going to be talking about research that we've done both at the University of Arizona and at the University of New Mexico, mostly in healthy participants. So you just heard about people who are in minimally conscious states. Now we're talking about research mostly on undergraduates who are mostly conscious states, and trying to modulate circuitry in the brain that's involved in mood and mood perception, as well as the default mode network, which is involved in internal states of rumination, mind wandering, and other types of off-task states. So most of the work was done here at the Center for Consciousness Studies in collaboration with Dr. John J.B. Allen, who's here in the audience. And I'll talk a little bit of some work I did when I was on postdoc. So usually in these talks, I have to give a setup about what ultrasound is, how it works. I don't usually have the world's experts to give me a setup. So I'm just going to go right into the work that we've done at the university. And Dr. Allen and I got interested in this technique back in 2013 when we ran into this guy, which I think most of you know. This is Dr. Hammeroff. He's the director of the Center for Consciousness Studies here. And Dr. Allen and I were actually doing studies with deep brain stimulation, where they do neurosurgery. They implant electrodes down into the brain of Parkinson's patients and OCD patients in this case. And they directly electrically stimulate the brain in the operating room. And so Dr. Allen and I were doing studies, 8-hour surgery sometimes, where we were in there trying to record experiments and really watching the power of deep brain stimulation to change people's lives, to change what it's like for their phenomenology, but also changing their behaviors, in this case, Parkinson's and OCD. And so we got bit by the bug of brain stimulation and are really excited about these techniques. Obviously, with deep brain stimulation, you have to do surgery. And so this is very costly and dangerous for the patients. And we were really sort of convinced by the technology, but looking for something non-invasive, like you saw today. And we ran into Dr. Hammeroff one day in the operating room in the hospital. I knew Stuart from his work on the orchestrated objective reduction theory. And Stuart was actually looking for a way to test this theory, which is basically an alternative theory to information processing in the brain that says that the microtubules, which are the cytoskeletal structure of the cells, are actually important for, and the sort of interactions on the quantum level in these microtubules are actually important for consciousness and how consciousness emerges or relates to the brain. And so, as you know, in classical neuroscience, we're really interested in the synapses and the conduction of information through chemicals and electricity down the cell and chemicals through the synapse. So, Stuart came up to us in the hospital one day and said, "Have you heard about focused ultrasound brain stimulation?" And we said, "Well, that sounds kind of crazy. What do you mean? You can modulate the brain with mechanical energy?" No, it's all electricity and chemicals, right? And it turns out that the microtubules actually have a resonant vibration. So everything in nature that's physical has a sort of base resonant frequency. Microtubules have a resonant frequency around 3 megahertz, which is right in the range of the ultrasound frequencies. They're typically used in the hospital to image the brain. And so Stuart, ever the pragmatist and experimentalist, was looking for a way to test the OR theory. And he's an anesthesiologist, so he decided to put an ultrasound up to his head in the hospital one day. That's literally Stuart's brain, you can see behind him. Now, of course, you don't get a lot of good imaging with ultrasound because the skull gets in the way. You can actually see a bit of his brain tissue down there. And he was actually really motivated to test his theory of OR by basically using ultrasound to resonate the microtubules. The hypothesis was if the microtubules have something to do with consciousness, you should change conscious state of the subject or the patient. So in the first study, prior to our work at the university, he basically took people with chronic pain. He did 2 or I think actually 8 megahertz ultrasound to the prefrontal cortex. And what you can see in the red bar here is that he was able to reduce self-reported pain in chronic pain patients. It wasn't actually significant. It was approaching significance. But for Dr. Allen and I, when we looked at this data, we were interested in the OR theory and helping him test it. But really, we saw this effect where chronic pain patients were actually reporting increased mood effect. Now, if you know anybody who has chronic pain, it's really hard to put them into a better mood. They're constantly perturbed by the pain signal. And so even though they didn't get a significant pain effect, they did get the significant mood effect. And Dr. Allen is one of the experts on depression and finding biomarkers for depression. And so we started thinking, well, this is kind of interesting. Could we stimulate the brain in people with depression or healthy people and actually change their mood state, so the conscious state of mood? Then, being a curious graduate student at the time, I started reading the literature, reading Dr. Bystritsky's studies, reading Seung-Schik studies, and got blown away by the fact that there's a whole field of ultrasound brain stimulation that was emerging at that time. And I'm not sure everyone agrees it's through microtubules as the mechanism. We can debate about that later. But we got really interested in this technology as a way to change the conscious state of subjects. Really as a tool for testing different brain networks, but also as a way to think about therapeutic interventions. And so we teamed up with one of the other founders of the field, Jamie Tyler, and a company that he had at the time called Think or Neurotrek. They had this little ultrasound device that you see here that my little brother Kramer is testing out and modeling. And by the way, his band is going to be our house band starting tomorrow. So Air Cool Jenny will be playing. And so you can see this is relatively simple technology at this point. It's a single channel transducer. And we decided to do a very simple mood study. Sorry, the scale is off there. But very simple mood effect study. So take people's baseline mood, try to target centers in the brain that we think control the regulation of mood and emotion, and then basically see if it changes the way people feel. It's a simple self-report study in the beginning. We didn't want to give a task because that could influence the way people feel. For our undergraduate subjects, you sit them down and make them do a 2-hour task. It puts them into a bad mood. And so we didn't want to perturb them. And honestly, we took their phone away from them. We did the first couple studies in the basement of the hospital, which is sort of a scary place to go. There's some old operating rooms and old equipment, and it looks like a horror movie. So, you know, within the context of that, we were trying to make people feel better. And basically, for a series of studies we did starting back in 2014, we targeted the right inferior frontal gyrus. So this is a part of the brain that deals with control, so usually behavioral control. But there was some evidence that it also deals with emotional control and regulation. And we know that self-reported happiness is increasing this area, regulation of negative emotion. So if I put you in a bad mood or give you some difficult stimuli, this part of the brain is going to activate to help you control that state. And that obviously affects how you feel over time. And then it's also a target for bipolar and depression. So basically what you see is we targeted with focused ultrasound right on the right inferior frontal gyrus. These are healthy subjects. These subjects are in the basement of the hospital, take their phone away from them and make them sit there for about an hour. And the basic effect in the placebo condition is mood doesn't change. So on this scale, up on the scale means that you're reporting happier mood states, less sadness, more happiness, just kind of feeling better. And what you can see is actually with 500 kilohertz focused ultrasound just for 30 seconds on in this study, we were able to make people, put people in a state where they reported a better overall affect or mood up to about 30 minutes. Now, if you think back to both Seung-Schik and Martin's talks, this window of change actually is really interesting to look at over time. And what you can see is our maximal effects were out at 30 minutes. And the people in the stimulation condition did seem to be happier with the self-reports as well. We did a little follow-up study to see if that network that we were targeting with the right inferior frontal gyrus was indeed modulated by ultrasound. So what you're looking at here is fMRI connectivity. So you're just looking at relative to the right IFG, who's it talking to? What kind of brain regions is it syncing up with? And the red are positive areas that are sort of moving together or talking together. And in blue are regions that don't like each other or are not communicating. And basically, if you look at post-ultrasound or post-sonication, now you see a bunch of regions deconnecting or communicating less with that brain region that we targeted. And it turns out a bunch of those regions are related to emotion control and emotion regulation. So there was only 9 subjects in this. It was a very small pilot study. But it does suggest that by targeting the right inferior frontal gyrus, we're able to change its connectivity, its functional connectivity with other brain regions. And that change is at least consistent with changes in mood regulation. Now, what's really interesting about this, I don't have the timing on here, but this is post 25 minutes. So again, this is not immediately after ultrasound. It's actually 25 minutes after 2 minutes of ultrasound in this case. And again, we see the largest effects out at about 25 minutes. So it's approaching that 30-minute threshold that Seung-Schik was talking about. So we see self-reported enhancements of mood. We've now replicated that in several studies. We actually used an imaging ultrasound like Dr. Hammeroff has used as well. I actually found slightly bigger mood effects with an imaging system than we have with the focused ultrasound systems. We can talk about why that might be the case later. And we also see altered brain network connectivity in the mood centers. But importantly, as I said, we see these offline mood effects, meaning effects after the stimulation is turned off, up to 25 minutes. We haven't actually looked further out than 25 minutes. We didn't expect to see effects happening this long in time. But now seeing the results that Seung-Schik has shown and others, you might actually expect to see these mood effects lasting for an hour to 2 hours. maybe longer depending on the dose, which is quite exciting. So we started getting really interested in these temporal effects, so how ultrasound is changing the brain over time, when I was a postdoc at the University of New Mexico. So we took advantage of a very simple paradigm where you can use a different brain stimulation device called transcranial magnetic stimulation, just a strong magnet. But if you put that over the motor cortex, which you see here, you can induce a twitch in the thumb. It's actually kind of freaky when you have it done the first time because there's a device that's controlling your motor control. But once the subject gets used to that, basically you can control the motor response on the finger, and you basically get an evoked response. Each subject has a sort of amplitude at the baseline that'll be at. And then what we did is we ultrasounded that piece of the motor cortex and tried to find out how long can we change the excitability of that motor cortex system. In this study, we actually used an imaging ultrasound system. It was the only thing that we had available with a very low duty cycle at 2 megahertz. And the basic effect is that right after ultrasound, what you tend to see is 1 minute, 6 minutes, and going back to 11 minutes, a change in the excitability of the motor cortex due to sonication. And then, of course, in the sham condition, you don't see a change. So this is pretty interesting because we see this effect up to about 6 to 10 minutes, but not longer in time. We're not seeing it up to an hour. That's potentially because we were using unfocused diagnostic ultrasound in this case. And if we would have used focused ultrasound, it's possible we would have seen a longer effect. But nonetheless, we do see an increase in excitability up to about 10 minutes, which is suggesting that taking the ultrasound off, you still see a change in the excitability of that system for some amount of time. This is a great paradigm to try to test these temporal effects, although it turns out that the motor evoked potential paradigm is really susceptible to the subject's state. If they think about moving, for example, it gets the motor cortex activated and it'll change the excitability of the system. So we got really excited about this because we wanted to try to map these ultrasound parameters. And it turns out it's not as straightforward as we had hoped. So we're still looking for other ways to map these temporal dynamics of the ultrasound effect. Other ways to do that is using behavioral measures. So when I was at the University of New Mexico, we also tried to look at how long in time behavioral effects would have been changed by targeting the right inferior frontal gyrus. So in this study, subjects are doing a really simple paradigm where they're supposed to push a button on every trial, and every once in a while, a stop sign comes up that tells them to stop. It's called the stop signal paradigm. And we know that the right inferior frontal gyrus is really important for that stopping behavior. So someone with ADHD, for example, is going to have trouble, and you'll see changes in that right IFG area. The question was, if we ultrasound into the right IFG, can we change the stop signal behavior? And does the change in that behavior map onto the change in the mood effects that we got? And hopefully we would replicate the mood effects in this study as well. Now, interestingly, what we see is a change in stop signal behavior, but only 5 minutes post-sonication in this study. It pretty much goes back to baseline between 7 and 10 minutes. So something interesting is happening with these networks when you use them for a behavior relative to when you let a subject just chill out in the lab and they're not doing a task. It seems like that network change is happening longer in time when there's no task relative to when you have the task. And that's something interesting that we can come to the discussion later, but it might really depend on how you're using these networks after you sonicate them in terms of the types of effects and how long these effects are actually lasting. So as I said, ultrasound disrupted behavior for up to 5 minutes. Interestingly too, if you look at the mood effects in this study, we did see subjects reporting more positive mood states, but it's not nearly as strong as the effects that we get when subjects aren't doing a task. So something's going on with the way the task is interacting with the mood states. While I was on postdoc, I got really interested in sonicating parts of the brain that are involved in learning mindfulness skills and mindfulness traits, and asking questions like, can we actually induce deeper mindful states when people are trying to learn meditation? and use that as a way to boost people into the meditation practice. A lot of this thinking came from Shenzhen Yang, who's here in the second row with us today. And we got really interested in this back in 2018 and trying to figure out if we could target certain areas in the brain that may either interfere with mindfulness training or areas where we could directly induce mindful states. Mindfulness is a very hard thing to define in the literature, but here's our definition. It means paying attention to moment experience with a type of openness and acceptance. Now, why would we want to use a tool like ultrasound to facilitate mindfulness training? Well, we know that mindfulness has all kinds of positive benefits if you stick with the practice for a long enough amount of time. It can induce all kinds of interesting, what they call unusual states. And some of those unusual states can be integrated into a person's life, coupled with training with mindfulness skills to really impact the way the person responds emotionally to the world, their resiliency, and actually impacting their physical health. It's also very useful as a treatment for many different disorders, especially chronic pain. And so lots of folks like us have been looking for ways to facilitate the practice to try to get these benefits out to the public faster. But this tool also helps us to study the different networks in the brain that we think are involved in some of these mindful states or unusual states. So it actually helps us to map those states as well and updates our understanding of how mindfulness is impacting the brain. We'll skip this because we're a little late here. So the problem is, if you've tried mindfulness training, is it's often very difficult. There are many barriers, especially in the clinic. If you have a patient with chronic pain, it's very hard to teach them to focus their attention on their pain. And with the promise that if they keep doing this for 30 years, they're going to be able to go into the pain and deal with it. And so one idea is we want to try to help reduce some of those barriers so people can get some of those effects from the meditation sooner and get motivated to maintain the practice. So there's a lot of different ways to think about how mindfulness is impacting the brain. We're really trying to induce what we call equanimity in the lab, which is that state of openness and acceptance to the experience. So if you think about something like chronic pain, and for patients who are dealing with clinical issues, this is definitely an altered way to relate to their experiences. And so the idea is to try to find networks in the brain that could either induce equanimity or perhaps interfere with it, such that we could elevate the baseline level of equanimity. And then they can train other skills in the mindfulness field so that they can acquire those skills much faster. So Shenzhen and I were sort of looking through the mindfulness literature and looking at all the different ways that mindfulness impacts the brain. And as you heard, one of the sort of really big benefits of ultrasound when you focus it into the brain is you can reach these deep brain targets. And so obviously the habit formation system in the brain shows up a lot in meditation studies because you're changing your habits, particularly some of those negative or maladaptive habits. And Shenzhen was reading about this really famous, really interesting disorder of consciousness called athymorrhea, which, to save you a long story, it's basically one of the most fascinating disorders of consciousness. People get bilateral damage to the pieces of the basal ganglia that connect to the prefrontal cortex, and it leaves a patient with this type of description that you see on the screen, a form of conscious awareness without any content. So consciousness per se. A lot of these patients will present to the doctor, they'll come in and it's very hard to diagnose them because it's not really clear what's going on with them in the beginning, but they will basically come in the office, they'll sit in the office without ever moving or talking, and they'll do that until you provoke them or perturb them. So when you say to the patient, hi, Jim, how are you? Jim will say, I'm great. And then you'll say, well, Jim, you've been sitting here for 12 hours staring at the wall. What have you been doing? And Jim will say, I've just been staring at the wall. Well, what's your experience like, Jim? Meh, the wall. Right? So these are very unusual people. They seem to be cognitively spared for the most part, but they don't have motivation to behave. It's a deep disorder of motivation. And one thing that's very interesting about them is that if they get into a situation where they're in pain, so if they put their hand on the stove, for example, they'll sit there with their hand on the stove until it burns, until someone basically removes that situation. And again, you ask them, what's going on here? And they'll say, well, yeah, my hand was really hurting. But, there's no suffering associated with that. There's no reason to remove my hand, so I just left it. All right. So if you're looking at this from the point of view of dysfunction, obviously this is extremely pathological. I'm not going to claim today or ever that we want to induce this in people. We would never want to do that. But thinking about equanimity, being open and accepting to the experience, these people are pathologically equanimous, if you want to think about it that way. And we started thinking about a biophysical model for equanimity and asking, can we induce a little bit of that? We don't want to make people athymormics, but if you can induce a little bit of that openness, that could go a long way for a lot of patients, especially if you're trying to train those systems at the same time. And so Shenzhen was the first patient 0, to sign up for this. And as you can see, like on the other slides, we've got neuronavigation. So we're targeting down to the head of the caudate nucleus in these studies. And the first question was, in very advanced meditators, can we induce something like a deeper equanimity state by targeting bilateral? So we basically had to move it from side to side, and we did about 5 minutes of very similar pulse parameters that you've seen in the previous talks. And to our surprise, not only did it seem to work in advanced meditators, it worked to a very high degree, meaning that it seemed to induce a very deep state of equanimity that some would claim was like 5 days of a meditation retreat or deep meditation training. This was quite surprising to Shenzhen and I, and we got very excited about this. Now, there is a caveat that what we're finding now in our work is that this seems to work a little better on long-term meditators than it does on people with no experience. That might be because long-term meditators have been working with these networks for, in Shenzhen's case, 55 years, something like that. So that's a lot of time of working with that network, using attention training to change the way that network is functioning. And when you sonicate into that network may be altered already before you do the sonication. Relative to some of our undergraduates coming into the study who just lived through the pandemic or living through the pandemic, who in our studies in 2020 and 2021, actually, we were some of the first people they had seen. That's a completely different human being with different experiences coming into our studies. But we did a small pilot study on long-term meditators. We gave them all kinds of meditation questionnaires, which are actually not that great at measuring mindfulness. But basically what we found is that we could induce more self-reported equanimity in people who were getting bilateral inhibition with ultrasound, or what we think was inhibition with ultrasound. And I just like to show this as a word cloud. I asked people to report a paragraph of what it was like for them when they go through these protocols. These are all of our long-term meditators, and the bigger the word, the more that they reported it across subjects. So you can see some sort of interesting words in there, like happiness, peacefulness. DMT showed up, if you know what that is, I'm not sure. I don't think we induced a psychedelic state in people, but it was in the direction of that sort of deep sort of body flow, I think is the sort of direction that people had that in. So when Shenzhen and I first started doing these studies, we got super excited about this because it beat our expectations. Shenzhen used to give me a 1 in a million chance of any of this ever working, and now I think he's pushing towards maybe 1 in 1,000 or something like that. So the data is updating our own prior model about how well we think this is going to work. So we came back to the University of Arizona and teamed up with Dr. Allen and an awesome crew of people here at the university and launched a lab called SimiLab, which we're now renaming to the Science Enhanced Mindful Awareness Lab. And we really started asking the basic questions about how well is this technology working? So the previous slides that you saw, there's a lot of caveats to getting ultrasound into the brain, focusing it to the targets, trying to figure out whether you're actually on target and modulating that system. or off target and modulating some other system, or is it just an auditory confound because you can hear it? So we wanted to come back to the university and really try to find ways to find these effects in these systems, and then try to see if we could change the dose to get bigger effects for people, but that are useful for their mindfulness practice. And so all the work is always going towards whether this is sort of a useful clinical intervention that impacts people's lives outside the lab. So we decided to go after the default mode network, another region of networks in the brain that really shows up with mindfulness training. And you probably know about the default mode network, but it's basically a network that we really don't understand all that much about, but has been very heavily studied in the neurosciences. And it shows up when you're in the MRI scanner, but you're not on task. So we put you in the scanner and we just say, stay awake, right? And different people are going to have different internal conscious states. Some people will ruminate. Some people are going to think about the war. Some people are going to think about the pandemic. Some people are going to try to meditate, right? People do different types of things. But this is basically the network that shows up when you're off task and sort of in that inner state. Now, if you have something like depression or anxiety, you might be more likely to ruminate in this state. So think about negative thoughts. If you're our typical undergrad, they usually think about homework or what they're going to do later or how stupid the study is, but they're usually thinking to themselves and sort of mind wandering. This network has also been related to the sense of self, and you wouldn't want to say things like this is where the self exists, but this is definitely a network that's organizing the bits of networks in the brain that are useful for self-processing, for self-representation and the information of the self. And what we know from lots of mindfulness studies is that this default mode network becomes less activated when you're meditating in the scanner. It's also altered from long-term meditation practice. And so it becomes a really interesting place to think about targeting with ultrasound. This is one really cool study where they actually were using fMRI neurofeedback. So they're using the MRI signal to feed back to the subjects as they're trying to meditate. This is over about 30 minutes. And what you're seeing is that as they sink into the meditation practice, you get less activation of the default mode network. So that's a pretty neat finding. Long-term meditators, you can see it sort of maps onto their phenomenology in a really interesting way. So the more down-regulated the default mode network is, the more sort of reports around concepts like equanimity are going to emerge. Now, if you take undergrads and try to teach them meditation that day, put them in the MRI scanner, it's not a very good experience. So they're trying to meditate, and now the default mode network is activated, particularly the posterior cingulate, and pretty much it's not a fun experience. So they're like, this sucks. I want to get out of the scanner. I hate meditating. I'm never doing it again. You know, that's kind of the state of these subjects. And that maps right on to their activation of this system. So when we're thinking about equanimity, thinking about being open and accepting as a state of experience, this becomes a really interesting place to think about targeting with ultrasound. And actually not just for mindfulness training, but you can think about for a therapeutic intervention in general. Helping a chronic pain patient be more open to their pain actually would likely help them deal with that pain state that they're having. So what we tried to do is mimic these types of paradigms, but ask, instead of using feedback, can we use direct focused ultrasound into this region? Attempt to inhibit that first prior to meditation. So what happens when we inhibit the system without teaching people meditation? If that works and we can find some neuroimaging evidence that we can change that network, what happens when we teach people to meditate in that state? So this is a series of studies we started in 2020 during the pandemic. It was quite difficult, but we recorded 29 subjects. And a very simple paradigm, again, we record baseline brain state with fMRI and lots of self-report phenomenology scales. So what's it like for you? What are you thinking about? How much ego processing do you have going on? So we just ask a bunch of questions like that. The subjects don't know anything about the default mode network other than we tell them we're targeting that in the consent form, and they're not cued into anything about meditation or how this would change their state. And then we basically do the ultrasound either inside or outside of the scanner, depending on the study. And then we put them back in and look at the brain change over time. And again, we were really interested in the acute effects, so right after sonication, but also what's happening up to about 25 to 30 minutes after sonication. The system that we're using is a little different than the ones you saw before, but very similar in physics. So basically, it's a focused ultrasound transducer. The beam is being focused down to the posterior cingulate, which is about 60 millimeters down. And we have to take a structural image of each of our subjects as well and guide that brain stimulation down into that part of the brain. I don't have time to show you our modeling work, but the skull is definitely disrupting the beam. And so we show you these beautiful beams here, but at the part of the brain that we're looking at, the beam is probably enlarging a bit and sort of being pulled back towards the skull. This is our lab manager and our graduate student, Brian Roller, in the lab. And Brian is doing some pilot testing here with his brain in the background. So the navigation systems look a bit like this. So, oops, I guess I'm not going to get a video for you, but basically that cone is the idealized ultrasound beam going into Brian's head at this point. And I think Brian is here somewhere. So to the safety point, he's fine. He can raise his hand. So thank you. He's had this many times. And so basically we guide this down in real time and we actually measure with each pulse. So as Seung-Schik said, it's pulsed ultrasound. We're going to measure each pulse and see where that beam is and try to see how the brain is changing for each of those pulses. That was the idea. Now, when we back up and look at the phenomenology, what I'm doing is I'm plotting one of the self-report scales on a radar plot. So 0 or no change is right in the middle. And then more change is going out to the outer rings. And really interesting for us, what happened is that stimulation relative to placebo, people started reporting altered sense of time. So about 80% of our STEM people reported that. Many of them reported deep inner peace, which was quite surprising to us. So these people are not meditating, basically getting paid to come into this study, and they're sitting in the MRI scanner for an hour. It's a pretty boring study. And overall, relative to placebo, which also had a bit of effect there as well, you do see a change in the phenomenology of these participants. Some people also said there's a dreamlike quality, which is kind of interesting. And actually, opposite of our prediction, they had a bit more thinking about past experience and sort of their thoughts were sort of happening more. We were thinking inhibiting the default mode network would reduce the amount of overall thoughts. Now, when you dig into the phenomenology of these subjects, it turns out that what was happening, at least on their reports, is that the thoughts were happening, but not sticking. So the stickiness of the thoughts was going away. And that's exactly in the direction of what we were hoping, which is more equanimity, less sort of inner stickiness around any type of processing. When we look at these effects, so placebo, 5 minutes post-sonication and 20 minutes post-sonication are here. You don't see much of a change in the default mode network. You saw a little bit of a change in the angular gyri, which is part of the default mode network. But 5 minutes and especially 20 minutes post-sonication, now we start seeing larger effects in the default mode network. These aren't huge effects, but they are definitely changes in the core networks of the default mode network. And what's really interesting to us is that those effects are accumulating over time up to 20 minutes. Now, if we had a little bit more funding in the study, we might have looked actually later in time. And the question is, would we have seen even a bigger effect? And then those post-self-reports that I showed you are always given after, so that after they come out of the scanner. So those are usually at 35 to 40 minutes, actually. Let me check my time. Oh, all right, well, I'm gonna skip this. This is another study we did where we tried to do a dose response. Always with these types of stimulation studies, dose response doesn't turn out exactly like we hope. The brain is a nonlinear system and it doesn't always respond in a linear way. But we basically tried to do different doses of ultrasound within subjects. So they came back on 5 different days. And the brain results are a bit messy and I don't have time to dig into them, but the subjective reports are quite interesting. As we increase the dose to 10 minutes, in green, you're basically seeing a stronger subjective self-report. Now, again, these are non-meditators. They've never meditated before, and these people have never done psychedelics, by the way. So They're not very used to these sort of altered types of states, and yet they're reporting more and more change in their inner subjective state over time. You can see there's sort of spacey, disconnected thoughts were flowing free. Overall, it was positive for all of the subjects, which is very important for us. We want to make sure that this is a good experience, and I forgot to show you here. Fear, felt afraid, felt suspicious, all of these things are not increasing. Quite important for these types of studies. And then lastly, we also looked at the individual changes in the default mode network related to the phenomenological reports. And basically what we find here on the bottom right, so in placebo, there's no correlation between brain change and self-report. On the right here, what we're seeing is that the subjects who had the biggest change, so the most equanimity report, had the biggest change in the default mode network. And particularly, the more that the nodes of the default mode network decreased their connectivity, the more equanimity report that we started seeing in these studies in those subjects. And so the biggest subjects equanimity report had the most disconnection in the default mode network. So that's a more exciting and more compelling result to us, because it suggests that what's happening in the brain is actually related to the phenomenology change, which should be the case if this brain stimulation is actually working. So the next step for the lab is now to start combining these interventions with a context, in this case with a context of mindfulness training. And so there's a lot that we can say about the set and setting, what Martin was talking about, what's happening in the brain before the sonication comes in. Also what's happening in their mental set. So the way that they expect, what they expect to happen to them, their expectations about these types of states, all of those things might influence the effect that the subject has. And so in some cases you want to control for that. In interventions, you might want to boost those types of effects. And so what the lab will be doing now is combining with a 2-week meditation course that's been developed by Shenzhen. And people get 4 doses of ultrasound throughout that course, targeting this default mode network region. The question is, if we can boost equanimity while they're training, do we then boost the longer term effects of meditation? So after the 2 weeks, do we actually see positive changes happening up to 2 months, up to 4 months? There's some other studies that suggest that that could be possible using psychedelics. We want to know if we can replicate that with ultrasound. Okay, so here's where all the work was done. Thank you so much for your time. I think I have time for one question. It is quite dry. Yeah. Oh, we have someone beating you, I think.
So do you believe or have any sort of evidence that maybe this could be used for other types of skill acquisition? Because it kind of occurs to me that skills fall in an S-shaped curve, where, you know, at the very beginning, you're so awful, you just can't do it. I think it's a linear section where you kind of motivate yourself, and then you have the discernment to, you're okay with the lower rate of acquisition.
Mm-hmm. Right. It's a great question. Shinzen's laughing because I think he,
Yeah.
yes.
Skill acquisition in general.
Yes.
All right.
Yes, you know, we're not going to claim that we found the equanimity center of the brain. The claim is that we're increasing some openness to the experience. If that's the case, and that's still something we're testing, then it should be useful for any type of skill acquisition. We're going to claim that the attention skills that we're trying to train in the lab are generally useful for other things. So if you're Steph Curry trying to shoot the basketball, you know, this skill acquisition of attention is going to help you with these other skills that you're trying to learn. So we're going to try to push you into mindfulness training anyway. But I think, yeah, if you're trying to learn something else, it would be useful as well. Yeah, quick question.
Just had a question on what exactly the end product is with the stimulant in terms of the experience. Is it something that opens the door to a greater reality? Is it an illusion? Is it simply feeling relaxed in a way they didn't feel relaxed before? And why is it that the stimulant is necessary for mindfulness? The tradition is in a lot of the Eastern religions is just solitude and concentration. So why do we need these artificial stimulants?
Great question. I don't think we would argue that the stimulant is necessary for the mindful state. As you said, if you have the time and the ability to sit, maybe go on a retreat, for example, hopefully you don't need the ultrasound for that. And that's why we've targeted very difficult cases, patients in the clinic, for example, first, who actually, if you teach them mindfulness, you sometimes get the opposite effect. You get an increase in their symptoms before you get to those effects. So this is just a boost for people. And the hope would be that you get a taste of the rewards of the practice such that you decide to go do this on your own without the device. That would be a win for us. We don't want you to have to use these things at home. We don't actually know about the long-term effects yet. Lots of labs are looking at that. So we would want people using this every day and relying on this as an inducer of their mindfulness. It's just a way to get you going.
So the end effect is simply getting calm or something beyond that?
End effect would be taking these skills into your life, I think, is where we'd like to get people. But different people have different goals when they sit down for mindfulness as well. So we want to help them reach those goals, but reach them in a healthy and integrated way. I think we have to move on, but we'll have a panel, so hold it. Maybe it'll ruminate and simmer. Okay, thank you for the questions. We have an excellent speaker coming up next, Dr. Bystritsky. We are very lucky to have him in person, and thank you so much for being here. Dr. Bystritsky has been in the ultrasound neuromodulation field and really been cultivating the field for the better part of about 20 years. He is now emeritus professor at UCLA and has a list of awards that would take about an hour to read, so I'm not going to read those, but has really been cultivating this field for a very long time and has really seen the field develop, and so I think he's going to give us a nice overview of that. So thank you for being here.
Okay, good morning. I will try to give a review of the field. A lot of stuff has been said already, so I have much less thing to work at. First, I wanted to say that I'm going to give a little bit also historical background of the field, and it has been a struggle, actually. It's an interesting new field. This is my current status. I'm a professor emeritus at UCLA. I'm on a requote, 10%. I'm also have been working in anxiety disorders program. I've funded this anxiety disorders program at UCLA almost 40 years ago, and OCD program and target brain stimulation, where we were inserting electrodes in human brain. I'm also founder and CEO of BrainSonics Corporation, that is a 20-year-old company, but I'm accustomed to think about myself still as a researcher. It's evidenced by my not taking any salary from this company and just actually contributing my own funds into that. So that's my conflict of interest, but I really don't have one except passion for the field. I'm also co-founder and board member of TBD Foundation, which is studying consciousness, and I'm the president of Institute of Advanced Consciousness Studies in Santa Monica. But I'm not primary consciousness researcher. I'm actually a psychiatrist. And I'm actually doing something like that 2 days a week. And the field has changed. if before we only used drugs and therapy, now we actually could use imaging. And I started in this area long ago, 20 years ago, conducting imaging studies together with Michelle Krask at UCLA and Susan Buchheimer, dipping into the brain and seeing what produces anxiety. We did a lot of provocation studies of anxiety in MRI. And actually, one of the first neuronavigation guided treatment of anxiety using function of MRI, but I always try to look for new tools. I think tools are very important in studying any field, and the better tools we have, the better findings we'll have. So as people said, focus ultrasound is one actually of the best tools around because it could go deep, it produces small focus, and it's capable, you're capable of changing it, like you heard from different talks here. And it can really reach midline of the brain and go into very interesting areas that are responsible for a lot of functioning in the brain. My interest was in trying to suppress amygdala directly because that's a center of anxiety. So the field started actually around 1930s by first application to the brain tissue, and it was done by Harvey. And then there was a renewed interest around the world in United States by Fry and Gavrilov, in France, Lele, and many people started to see that it actually can affect the area. In 19th, Dr. Frank Joles from Brigham and Women's Hospital, who was very, very advanced person. He always went after new tools, and he basically was behind the inside tech production of focus ultrasound, where people burned holes in the brain. It's really a surgical instrument, but it's brilliant because it uses multi-transducer device and could get into any area of central brain and actually eliminate, for example, metastasis of the brain. I started to get into this area in 1999 because I was not satisfied with cognitive provocation and suppression of anxiety. I wanted something more, something that is compatible with MRI. So I started to read, being originally from Russia, I started to read basically Gavrilov's papers and Cyrilnikov, and realized that this is what we could use. I filed the first patent, and I'm going to give you anecdote. UCLA refused to file the patent because they did their own search and they could not find anything. They say, we're not going to spend any money on that. And that was my experience for the first 10 years working in this field. Basically, I had to buy the patent, open the company, raised a little bit of money, went to Brigham and Women's Hospital, where I met with Joles. And then I met in the same place with brilliant scientist, Seung-Schik. We liked each other, and we decided that if we have money, we need to do experiments. And we started to do experiments on animals. And the first experiments were spectacular. We also did some experiments with Kullervo-Heiden on sciatic nerve of bullfrogs. And that gave me first a notion of the power of ultrasound, because we did that around 2006, and suddenly we were able, for very short stimulation, inhibit conductivity in sciatic nerve for 45 minutes, fully reversible. That was drastically different from RTMS, where you stop, or DBS, where you stop electricity and the effect stops. You need a lot of repetitive. Here you have this after effect that lasts sometimes for hours. So we realized that we are onto something very interesting here. So, and then all studies were under radars because we could not publish that initially. And then Jamie Tyler, who is also a brilliant scientist from here, he published first papers on using low intensity focused ultrasound in brain of mice, and in slices of mice brain first, and then he was able to move the tail, which was a very interesting picture. You could actually jerk the tail of the mice with focused ultrasound by stimulating the brain. Those data were later replicated and then later criticized by Caltech because they thought that it's acoustic. And it may be those were acoustic, but we have now other evidence that it's not just acoustic effect that we see. So the first interesting imaging study was done on rabbit. It's kind of, I think it's classic, done by Seung-Schik and was published 2011. You shine some light in eyes of the rabbit, and you get visual cortex stimulated. And then you could suppress it with only 9 seconds of ultrasound. You could suppress it for 11 minutes. So suddenly cortical area, visual cortex becomes unresponsive. So that led to actually next study by Seung-Schik and his group, where they did inject rats with PTZ and were able to stop seizures. And it worked. We have actually recording of that. It's amazing. It's almost like you're giving the animals shot of Valium. So, we also started to do experiments at UCLA, where we used mini pigs, and we were changing blood pressure of mini pigs using focused ultrasound. I received at that point a NARSAD Distinguished Investigator Award, and we could augment and do some anesthesia studies. But the struggle was actually applying for an IMH awards, because you basically apply for a pilot award and they say, where is your pilot? And then it's being reviewed and they say, the reviews were totally ridiculous. It was like, it cannot work because it will not work. I hope you were getting the same reviews for some of your studies. That means you're on the right track. So basically, studies were saved on animals. I was invited by Brain Stimulation to do the review in 2011, where I actually reviewed all the studies, and my conclusion: we need to go to humans. There is no way we just can continue doing animals. Let's go to humans. And another brilliant scientist, Dr. Stuart Hameroff, just did that, as we could see in 2012. And and showed some effect that was very encouraging, that helped us actually to get our first-in-man study in epilepsy through Human Subject Protection Committee, because we could not do that for a year and a half. So, and that followed by a lot of other studies by Dr. Yu kind of gave brief review of his studies, and then Legon and Tyler did several studies on somewhat sensory, and also Dr. Yu did that. And we at UCLA, basically I was not allowed to come near the patients because I had conflict of interest. It was conducted by all our PIs. Dr. Monti started to do consciousness study, and UCLA IRB permitted to do that because those people have nothing to lose. The same thing was for extensive Alzheimer's disorders and epilepsy study. The first in men actually in functional MRI was done by Dr. Stern and his group. Here is a very interesting picture. This is very dear to me because this is the first epileptic patients. And we were stimulating temporal cortex through the temporal window. And then those patients would go, it was a very interesting study. It was guided by FDA. We tried to persuade people that it's safe. So it was, we would stimulate the area, and then this treatment-resistant epileptic still would go for surgery, and we would get the histology. So this is basically, we could see the place where we actually stimulated in distant areas, like in frontal area that responded to that. We recently published this study, actually. It took a long time because we also moved to higher intensities. Focus ultrasound, I'm convinced, is just like any other treatment, it's dose related. So we were allowed to go only under 720 milliwatts per centimeter square, which is normal ultrasound threshold for Doppler. And we were able to go 10 times higher, and still histology was good. So we just recently published that, and we're going to publish a bunch of other safety studies on that. Mechanism of function. It's going to be debated again. Microtubules, mechanical receptors. Here is a list of to me, what you're doing, you're producing mechanical energy, you're distending the membrane, and it changes properties. It's maybe letting ions going back and forth, hyperpolarization, but definitely what was discovered in Seung-Schik experiments in Harvard, that there are activating parameters and inhibitory parameters. This is actually going to be published, and some of the co-authors of this, I'm like the last in this group. It's a publication from Institute for Advanced Consciousness Research, and director of institute is here, Nika Regen. All right, and you participated in that, Jay participated in that. So we're describing safety and different mechanism in this. It's going to come probably in a couple of weeks. So this is BrainSonic's device that was used in most human studies. So it's basically, it's a hockey puck, which is transducer, and it goes right under the coil, and MRI, it's fully MRI compatible. So you could see where you kind of hit and how other areas responding. Again, there are a lot of things to figure out, like how to do this through the skull, but we're working on that. We're working on multi-transducer device, on specific pads that could modify the focus. And one of the inventions is, oops, I don't know, it's starting to move. One of the inventions is these pads. Basically, we develop pads that permeable by ultrasound and those that block ultrasound, and they're indistinguishable. So this just gives you an opportunity to conduct double-blind trials in very elegant way, and we're starting. So device right now in 14 different centers around United States. Some of them have 2 devices, like Harvard has 2 devices, UCLA has 2 devices. And we already, these centers, first of all, now, as Jay said, suddenly areas start to get speed, especially the last 2 years. People in these different centers starting to get NIH and VA grants, and my understanding that all these centers combined received last year over $10 million in different government and foundation grants. They're studying different locations, different paradigms, and dear to my heart is OCD Breakthrough Award that Wayne Goodman and Darren Doherty won, and they're about to start double-blind placebo-controlled trial for treatment-resistant OCD. And we about to start at UCLA, where, first of all, we already started GED trial, and first patients have been recruited in Medical University of Southern Carolina, and Coma trial is about to start. It received a breakthrough pathway from FDA, and we hope that within 2 years we'll have some treatment for this population that really has no treatment. 50,000 people in the United States at any given time are in coma. So this is badly needed. So we used it in MRI, but we also have targeted neuronavigation. That's how it looks like, and this is some of the results that coming out right now. So first of all, this is from Bardan and Mark George. This is brain stimulation journal, was published last year. You could see clearly different targets that they hit. They did a placebo-controlled trial on a pain threshold and pain recognition. This is the results. Participants required higher thermal stimuli temperatures to indicate pain and show higher tolerance after TFAS. So this gives us a thought that maybe we could modify pain right by going to this thalamic area. Another study is done by on normal controls in the function of MRI by Taylor Kuhn at UCLA, and it's aimed at amygdala, which is center of anxiety, but it's done on normal controls. It's So the results are here. You could see activation in correct area. That's targeting verification by a cell. And this is preliminary data on these 16 people. They used also controlled other target. They used intrarenal cortex. So you could see here group finding on ASL in amygdala area suppression. So, as I said, there are a lot of recent publications. The last year, only UCLA group published 4 different clinical studies. But interestingly enough, we're starting to give this device to trusted groups in the communities that conduct clinical work, because we want to accumulate more of open results. These studies are being done under IRB. For example, we're collaborating with neurological associates of Los Angeles, and they treated 2 groups of very treatment-resistant patients. One is GAD, and those people went through everything, including some of them ECT, but several rounds or RTMS, all the medications and so on. So some of those patients were referred by me, so I know exactly. I treat very treatment-resistant GAD and OCD. So this is the results on the first patients. 13 of 16 really were responders. And you could see only 3 did not respond. And this is by Beck anxiety inventory. The same results were obtained. Actually, 1 patient responded by Hamilton. So with OCD, this is very interesting. This tells you that maybe the future of psychiatry will be actually applying this energy to specific neuronal circuits. That's actually how I imagine that's probably going to be done. Assessment, MRI, EEG, and then we will know the circuits, and then we will be applying this kind of treatments. This was interesting study, and Seung-Schik, you were responsible for that study, because we heard about that you're increasing this state, right, of kind of no thoughts in the brain, right? Meditation state with, and that was from head of nucleus caudatus. So we decided then to use that. Okay, that's a great treatment for OCD. So we aimed at that location, and this is the result in the first 6 patients. Didn't work, it worked in only 1. We were so disappointed. Then we spoke to Wayne Goodman and other people and said, why don't you go to VCVS where you insert electrodes for DBS? And it's completely different results. So it's actually controlled by location. It's an open study, but it's controlled by location. Some of the patients participated in this second study who were failures in the first one. But it's, again, very treatment resistant. It's like really pre-surgical people, and then they suddenly, some of them start dramatically respond. This is basically statistics right now. I just run studies. Before, like 5 years ago, you could find maybe 10 studies in this field. Right now, it's 400 plus per year. So and this year just started, and it's already above 200. So the field is growing rapidly. Focus Ultrasound Foundation publishes on funding, and they have about the same graph. So the field is really developing. So, but let me speak a little bit about the future. This is, to my knowledge, most promising applications. One is, and it's relevant to the area of consciousness. You could use it for brain mapping and maybe to figure out neurocorrelates of consciousness, because it has powerful suppression. So you could suppress certain areas of the brain and interrupt information flow and maybe change things in the consciousness. And I think, Jay, your studies definitely demonstrate that by modifying this default networks, right? It could be used for neuromodulation. So it could be the next big treatment. RTMS took a long period of time. Interesting fact, it takes so long to develop this treatment. First patent for RTMS was filed by De Beer, who was colleague of Freud in 1903. First use of RTMS was 1945 by I forgot the name of the English guy, but then it's only in 2008 there was first FDA approval for that. So for focus ultrasound, it's already takes more than, no, it's close to 100 years from the first use and 20 years from the first patent. So, but it's going to happen. We're starting several pivotal studies for OCD, anxiety, and coma. Hopefully, we would be able to give this to people. It also could be done outside of MR with neuronavigation. We're collaborating with every neuronavigation company and precision within millimeters. And we validated that, and I think it's going to be better than or good in combination. It's not very good for the cortical areas. RTMS is good for certain cortical areas, so they could be combined actually. In fact, on one patient, anecdote, again, clinical anecdote, it was treated with suppression of amygdala. And before that, he had 2 runs, he had anxiety and depression, 2 runs of ECT, 2 runs of RTMS, 20 different trials of the medication. So after suppression of amygdala ultrasound, he did not respond. His anxiety got better, depression got worse. Then he was treated in the same clinic by RTMS, regular standard RTMS, and he miraculously recovered, and he's in remission for 2 years. So go figure. I think another thing that would be very interesting is brain-computer interface, and Seung-Schik already started experiments on that. Another interesting, we were proud that we're not opening blood-brain barrier in first animal studies, but actually we could open blood-brain barrier, and McDonnell and Brigham Women's Hospital group actually working on that, but we're also working on that, and we're trying to deliver currently biologicals in the brain, like for example, exosomes that could carry mRNA, and they could carry also different microdoses. For example, they could carry microdoses of psilocybin. Wouldn't it be nice to deliver those to specific area of the brain and maybe have less overall side effects, but maybe more targeted treatments? Maybe we could activate some drugs within the brain in specific locations and deliver those thousands of different drugs that are on the shelf right now because they did not get into the brain. I think this field expanding, imaging is getting better, our algorithms for imaging getting better, multi-transducer precision getting better. So I'm very excited to follow that within the next 10, 15 years. I think it's going to bring very good fruits to and maybe change as we do psychiatry from assessment to the treatment and contribute to neuroscience. That's all I can say. I think this is the future. Questions? Okay, I think we have a panel discussion now, or we're doing.
We have about 45 minutes. Maybe we'll go to general questions first, if anyone has them. Is Seung-Schik and Martin still out there in internet land? Maybe.
Yes.
Hello, thanks for sticking with us. Maybe we'll take your general questions first. We want to hear some feedback from you folks and see what questions you have, and then we can panel after that if there's time. So, questions you have. I know there are a few out there.
Hi. I have a general question, I guess. So I haven't really seen in the work that you're talking about kind of direct comparisons between the focus ultrasound and like the TDCS or TES methods to compare, at least to the cortical sites where you can look at both. Has there been direct comparisons to look at the effects on like EEG or the resting state MRI connectivity or the kind of activation effects?
That's a good question.
I don't think there were.
Yeah, there was one study I saw in 2018 or 2019, right before the pandemic, where they were comparing transcranial magnetic stimulation to ultrasound. I haven't seen it published, and I'm guessing it's because the pandemic has put a slow to that. And that was in a rat model of depression, and they were showing ultrasound worked. The effect size was about a third stronger, actually, with ultrasound. But in the rat brain, you have to understand it's sonicating the whole brain. It's such a small brain.
Right.
So TMS the whole brain versus ultrasound the whole brain in this model.
Well, like for you, I was curious, have you thought about comparing with the PCC work? I mean, I guess you can target that too with like TMS, TDCS. Can you see if, I mean, it'd be nice to show that there's some more effective outcomes with this, because I understand the advantages for deep brain structures, but it seems like more basic work needs to know the relative strengths and limitations if we're talking about also using it for cortical structures.
Yeah. I think all of those different brain stimulation techniques are legitimate and can be working together. Some of them doing better job focally, some more generally. I mean,
that's kind of my question, because like if you're a user and you're thinking, hey, which am I going to choose for this particular research question I have?
I think it's the next step, because right now we're trying to understand how every one of these techniques works.
Yeah.
Because TDCS has very many questions.
Right.
I mean, how the energy gets into the brain is one of them, because the skull is very good isolator. So it must be going through some other channels. Magnetic field is also not so simple. Tony Barker, actually, I remember him. He was giving a talk on a recent clinical TMS meeting, and he was showing some data that it's not, you would think that skull does not modify RTMS, but it does. And it's not all kind of clean-cut picture. So I think there are a lot of things that needs to be done on basic science, because we still do not understand how RTMS precisely work. And it has been on the market for...
Right. But at least we know with the electromagnetic stimulation techniques that they are targeting the signals that we know are the direct neuronal propagation signals. And so, I mean, I like all the ideas about the mechanisms for focused ultrasound, but they seem like they're more indirect, or we're not sure. So it'd be nice to know if like one is more effective because it's having a mechanical, impact. The others in other situations are more effective because of an electrical stimulation impact, things like that.
Well, what we know right now that it's every different location gives different effect. So, and I think your question is very broad. We need to narrow it down and answer the questions sequentially. In that.
I think that's a great research program. Hopefully you guys are working on it because it sounds really exciting. Thanks.
All right, other questions? Yep.
Well, thank you for these wonderful presentations. I have a question about mindfulness, equanimity, and ultrasounds. What is, in your opinion, the endogenous or natural equivalent? Sounding a bell, chanting or reciting a mantra, a chorus. So I'm wondering what could be the endogenous or natural equivalent of ultrasound stimulation in terms of mindfulness or meditation or equanimity?
Really good question. I think it partly depends on the mechanism. So the last thing that Sasha was talking about. We think that the pulse parameters that we're using are likely disrupting the default mode network integrity. So meaning when you go into an inner state, let's imagine you're worried about the war and you're sort of ruminating on that. Those networks need to maintain their activity together. They need to communicate information. And we know that mindfulness training might disrupt that as well. So focusing your attention on your breath will disrupt that inner process or that inner task. That's one hypothesis about what the ultrasound may also be doing. And when you couple that with mindfulness training, then you can switch to that mindfulness task a little bit easier. So we think maybe we're helping people switch in and out. And if that's the case, I think lots of different training techniques, so mantras or anything else that's allowing you to sort of switch out of the inner and sort of switch to the other task would be helpful. But that sort of depends on what the ultrasound is actually doing to that network. So yeah, it's a good question. Okay, we have some online questions. Seung-Schik and Martin, I wanted to ask too, do you know of any comparison studies or have you seen that TMS to ultrasound study?
Not that I know of. Can you hear me?
Yes.
Okay. So I think there has been separate studies investigating the effect of TDCS or TMS on functional connectivity, but I don't recall there's any direct comparisons. Probably there's one rodent study that you mentioned, Jay, but I think eventually we'll start to merge. But one of the things I think it is one of the questioners from the audience. I mean, you can probably stimulate the cortical area among all these different modalities and compare these effects. But for instance, PCC, I don't think TDCS can ever reach those areas accurately. I think the TMS will be even challenging. And just want to make sure that the TMS or TDCS, you have to think about the pathway that to reach the target. It's probably going to stimulate all the other areas in its path in terms of TDCS and TMS. Ultrasound, however, it's a focus, so it is relatively unscathed and untouched on its pathways compared to other modality. So it's more focal. So PCC, I think the ultrasound is probably the unique method to do it.
Yeah, that's a great point. There's something called deep TMS where you can try to target the posterior cingulate, but with TDCS, it would be unlikely you would reach that target. Also, thinking about, I was talking about the modeling that we've done and others have done, showing that the skull at certain areas of thick skull, so it's quite thick in the back here where we're going through, it's likely pulling some of the beam. So remember, it's that focused pencil-shaped beam. So we're pulling back. In the case of facilitating what we're trying to do in the lab, we might be actually modulating some of the precuneus, which is the next target in the path to the PCC. In our case, that's okay because that also shows up in mindfulness training. But I think when you're developing interventions, we have sort of error bars on where we think we're actually getting the sound. And you have to consider that path and whether you're actually modulating in that path as well. So I guess for the amygdala, I guess maybe you're going through the right temporal window. So that's sort of right prefrontal.
And thalamus too. So those targets are easy because they're right in the way.
Yeah, anything that is a straight shot from the temporal window, like thalamus. Luckily, there's a lot of things that are a straight shot from the fitting in the temporal bone. So that's helpful.
Seung-Schik, can I ask you a question?
If I can answer it. Please go ahead, sir.
On the record. So you showed that average of intensity was 4.1 and you went all the way to 14, right?
Right.
So
the 14 was the free water, which means there's no skull whatsoever. And then after you transmit skull, and it's based on the numerical simulation, we think it is pretty accurate. And it went down about 30%.
Sorry. Do you administer that amount of intensity on the skin and skull?
So skin, so you have to think about the intensity per area. So 14 watt, 13 watt is not really on its focus. So at the skin, it would be much, much less than what it is. We didn't even calculate for the number because there's no heating issues or whatsoever. So we believe that the intensity is very low at given area.
Of course, of course. The thing is, what we were struggling with FDA because they don't particularly want to administer that even on the skin and skull. They want to see the data if it's safe and if it's so they still go for an SR usually for 720 milliwatt per centimeters. So we did a study on open brains and we blasted them with very high energy and we found that there is no damage until you go above 24 watts. So even 14 is really supposed to be safe. I think that's the next few steps. We need to figure out if, and in an epilepsy study, we went all the way to 6 watts per centimeter. So I think the truth is somewhere between 7 and 14 watts. So you're on the right track. It just I was very pleased to see that.
Right. One of the things I like to add in the same context is, so therefore it is very important to run a numerical simulation. That is also backed by actual measurement, like the skin. You can actually measure the skin temperature using an IR camera. But just derive actual absorption of energy at given area. So all those numbers that I showed you today is based on this focal area. So therefore, if you are using this beam, the cylindrical beam of ultrasound that are going through the skull, it's much, much less. So the numerical simulation also helped, not only in terms of estimating the acoustic intensity, but also probably it's good to monitor. So as Jay mentioned, if you're sonicating through the thick skull, it tends to shorten the focal length. If you run the numerical simulation, you can actually see that in semi-real time. So in order to compensate that, you can actually put it a little deeper so that whatever the shortening of the focal length can be compensated for.
Right. By the way, we did study on 14 normal controls on the scapula, up to 14 watts, and we measured temperature on the skin and any side effects, and it's in live people, and we found no side effects. They did not complain of anything. They couldn't feel anything. So again, I believe intensity will be important for the future.
I have a couple of questions from online. If Martin was there, I wanted to ask a quick question about the patients. Still there? If you can say, what's it like for the patients when they wake up? Is it similar to what it's like when they wake up without any type of intervention? Because I know these patients wake up on their own.
It's hard to tell. It feels like it from the outside. Mm-hmm. The kind of behaviors that we see are the same things that you would see if you had someone recover spontaneously. In fact, in sort of as a stressed in the acute patients, we're having a hard time being firm about whether what we're seeing is spontaneous versus ultrasound. Which, makes complete sense scientifically, but also tells you that we think we might be able to confuse the two. So in terms of the third point observer type thing, they look pretty similar. From having done ultrasound to thalamus and globus pallidus in healthy volunteers, we've not been able to get consensus phenomenology about it, but we're also not sort of looking at it in a standardized way, for example, the way you are doing. So,
gotcha. Mm-hmm. Gotcha. Thank you. I mean, the thalamocortical prefrontal loop is involved in arousal overall. So, the overall level of arousal and consciousness. So I didn't know if they wake up a little more awake, so to speak.
Okay, let me here's an interesting datum that we keep seeing. It's not formalized, but we keep seeing it. So we do ultrasound. Patients typically seem just extremely sleepy the day after, the 2 days after at most. So it seems to me we're clearly messing with their arousal because they tend to become hypersomnolent. In fact, to the point that we have trouble the day after, and sometimes 2 days after, we have trouble doing the clinical assessment because they're always too sleepy. It could just be that we're hustling and bustling around them, and that fatigues them. We need a double-blind clinical trial, a sham-controlled clinical trial for doing that. But we're clearly messing with that, and then a couple days later, we see these new behaviors emerge after they sort of recover. So it seems to me that we're clearly messing with their arousal, but I don't yet have a sort of scientific number to show that. Yeah.
Thank you. We have a couple questions from the audience outside, and then we'll get to yours. Cedric asks, are the state dependency effects, as with TMS, for example, modulation can inhibit or excite depending on the current state of the networks. So I think he's asking for the state dependencies and what's happening before the ultrasound comes in, and that's going to affect the effect people see. I don't know if you want to.
There is that paper that just came out from Elisa Conofago's group, showing that not necessarily about, well, at least at the level of rhythms, right? A brain that has faster spontaneous rhythms when you intervene with ultrasound can have up to a 7-fold magnitude of response compared to a brain that has slow frequency, greater predominance of slow frequency fluctuations. So if by state, if we're happy to include in state just the idea of fast versus slow power spectrum density, that I think that works. It'd be super interesting to see whether you can have somebody think about a certain topic while you're ultrasounding, and if that leads to the same effects that people have reported in TMS. But I think that's still to be fully assessed.
Yep. Seung-Schik, I don't know if you wanted to add.
I think it is. I'm totally with Martin. Neuronal circuits are circuits. That's why we probably call it circuits. And a lot of neural communication, by definition, is state dependent. So further study is needed, but I believe there should be some dependency on the neural arousal state or the excitatory state. Mm-hmm. And I think it will be depending on where you stimulate them.
Yep, I agree. So with RTMS, for example, it got its first FDA approval for depression in 2008, and they were not even using neuronavigation. They usually just measure to a spot of the brain called the dorsolateral prefrontal cortex. It turns out if you do RTMS in time with the endogenous alpha oscillations, which are really important for inhibition in the prefrontal cortex. So for each individual patient, you time the RTMS to the peak of the alpha wave. You get bigger clinical effects. So it's definitely true in the RTMS literature that coupling to the ongoing brain oscillations is going to give you a bigger effect. Whether that's true for ultrasound, we don't know yet, but I think we would all bet on it, knowing what we know about brain circuits. Okay, go ahead.
There's certain bad states that are possible with meditation. I'm thinking dark night of the soul kinds of things. And I guess I've a question, the flip side, really about the same thing. I guess, have you seen any evidence that if you're stimulating the brain that you could actually induce one of these less desirable states? Or what is more likely is somebody who's actually already in a dark night of the soul, could you probably get them out of it by doing stimulation?
Yes, we have a whole workshop at 2 p.m. about that with Daniel Ingram. So yes, come to that. Well, we're going to talk all about they call them adverse events. That's the term now they're using in the field. And with meditation, it's not just dark night of the soul, which is at the extreme end, but uncomfortable situation, pain, fear and trauma, anxiety, all kinds of things can come up when you're practicing meditation. So it's not all good, although if you read the popular press, they make it sound like it's all good. And now we're talking about facilitating that with ultrasound, with psychedelics and other things. So you would want to be aware of those and be able to have a context and a container for helping patients or subjects deal with that. So we haven't seen it so far because we haven't done a bunch of long-term trials. But I think as you increase the dose, for example, of ultrasound and increase the dose of meditation, you're more likely to see these types of things emerge. And then what we do is we prepare the patients for that. We don't want it to be a surprise if things like that happen. And then if they do, you want to have, just like in the clinic, if something happens, you support that person through that process. And then it can actually be into the benefit of the patient over time. If you didn't warn them about it, then you've, if it just popped up, then you have trouble. Whether you could knock someone out of one of these severe adverse events, in the clinic, they have used TMS, RTMS, to try to reduce manic episodes. So one of the side effects of RTMS for depression is that you can induce mania. It's very unlikely, but it's happened in the clinic. And there's been a few small trials where they've actually, so you take the TMS to the other side, try to inhibit the right prefrontal, and you can actually reduce the manic episode. So it's possible something similar could happen for dark night, but we haven't tested that.
All right, thank you.
Mm-hmm. Other questions?
So there was some mention on the last presentation of the blood-brain barrier, and it just occurred to me that I'm sure it's useful for a lot of different reasons, but how are we making sure you're not targeting that, Jay, in your lab? Because that seems like it could potentially be very problematic if you're taking medications or things like that.
Yep.
Blood bearing with the intensities and with duration of the treatment right now, which is under 10 minutes, we try to monitor blood-brain both on animals and in humans, and it's not opening that. In order for you to open, you need to administer that for a longer period of time, about 45 minutes. That's where it starts opening. So we are aware that blood-brain barrier is there for some reasons, and also some substances, like some biological substances, you could get there by different additional techniques. So, and people working on that right now. But the way we're using neuromodulation is not opening blood-brain barrier.
And I would like to just add quickly on that issue. So traditionally, blood-brain barrier opening using focus ultrasound, you have to inject a microbubble agents to amplify the local effect of acoustic pressure wave. So what it is you inject ultrasound contrast agent. It looks like a milk. It looks whitish liquid. You inject IV and it goes through the bloodstream. It has a little microbubble. And then when it is hit by ultrasound, it wobbles and it amplifies the local pressure wave, and then it disrupts the BBB. That's the so far that's the people believe. And without the injection of microbubble, it's extremely hard to give that much of pressure, deliver that much of pressure to the local brain area. So for the given all the intensity that we've been using, it will be very difficult to break the blood-brain barrier. It's nothing to do with the duration of sonication. You can sonicate all day, and you may not induce BBB disruption.
I beg different. Yeah. Well, we just completed a study on rats with its collaboration with City of Hope in Los Angeles, and 45 minutes to an hour at even current intensities do open blood-brain barrier. So I think it is a matter of duration, and I think it's probably because of the pulsed ultrasound delivers a little bit more mechanical shaking there, but I could share this data with you.
I think if that is true, this can be another independent finding.
Another what finding?
It could be totally new finding, because that's a little different thought of school of thoughts. So it will be more depending on the pressure wave that has been created by injection of microbubble. But if the longer durational pulse, otherwise it will be not opening the BBB. But if it give it a little longer and then create a BBB opening, that itself has a meaning.
Yep.
Yeah, the peak pressure. So this is a sound wave that's moving through a medium, and there's a pressure wave associated with that sound wave. Typically, these peak pressures are below a megapascal, which is the measured pressure on the Earth. And when I first started doing these studies back in 2013 with Stuart and Dr. Allen, I was really worried about opening my own blood-brain barrier. So, you know, I think in these types of cases, it's good for the scientists to be worried and do the due diligence before they do it on other people. And it just turned out the University of Arizona, there were some folks trying to use blood-brain barrier opening as a delivery for Alzheimer's medications. And so I went and hung out with this group for about a year as Jamie Tyler was convincing me that this was safe to do it myself. And I actually went and watched those studies. And what happened is they actually tried to open the blood-brain barrier at about 0.6 megapascal, which is about where we are in the range of most of our studies. And they couldn't get it to open. So they wanted to open it without the microbubbles because it would be much more scalable. If you have to do microbubbles, it's a whole thing. And they couldn't get it to work. And so I watched those studies and I thought, okay, this is making me a little more convinced. You have to get these microbubbles and shake them violently. They explode. And then the other interesting thing, and I'm not sure if you saw this in your study, but what happens is it doesn't permanently damage the blood-brain barrier at lower intensity ultrasound. So the T junction, I believe is what it's called, this little protein that kind of binds the blood-brain barrier pops open. And over time, the blood-brain barrier gets repaired, which I think is amazing. Biology is such an amazing process. And if you do lower intensity, you don't actually damage it over time as far as we can tell. And so it was those two facts where I started thinking, okay, I'm going to do this on myself. So
there is even more to the story, but I can't report it.
Okay, good. Well, there's more to that. But yeah, this is the state of the science where we're at so far.
That it's safe. For now, I think thousands of treatments have been administered to the brain at current intensities, less than 10 minutes, and practically no side effect.
Yep. We're working on a paper reviewing over 400 subjects that we've done and no adverse events. Other questions? Sure. Oh yeah, you had one. We never got to it.
Going back to the first paper that I read about this subject, the one published in 2013 by Professor Ameroff, I remember there was a connection between transcranial ultrasound and the OR-core theory and resonance of the microtubules. So I was expecting today to hear something about this, or if there have been other studies trying to connect the ultrasounds with the OR-core theory and the microtubules.
Yeah, Stuart got funding to test other avenues of the OR theory. So we tried really hard to figure out a way to isolate the resonant vibration of the microtubule from the other proposed mechanisms. So shaking of the lipid bilayer, the ion channels, you'd have to sort of separate those mechanisms and try not to affect those while affecting the microtubules. And it's very hard because the microtubules are the cytoskeleton of the cell. So you have to go through all the other medium to get to them. So if you have ideas about how we could isolate that mechanism, we would love to test it, but it was quite difficult. I think probably the best way to do that would be organoids or some type of neurons in a dish where you can try to separate down into the tissue. There's actually a researcher who I think is here named Anirban who's been doing some really beautiful work trying to make very, very microscopic ways of measuring the insides of the cell. And so one idea would be to use really low intensity ultrasound, modulate the inside of the cell, and try to see if you can affect the cellular dynamics. The problem with that is you can't ask the cell how it feels. So whether you're changing consciousness or not on the cell is going to be quite difficult. So yeah, but Stuart, I think later in the day is going to be talking about some of the other studies for OR testing. So my feeling on the mechanisms is that it's probably multiple of these mechanisms. And so it's hard to isolate any one of them to see how that's affecting the system.
It's biological dynamic system, even cells. When you're taking neurocircuits.
Right. Yep. Other questions?
Jay, can I ask a question to you? I
Sure.
just had a question about how different is atymoria from things like abulia? Sort of, does it have any particular difference? You described it as both sort of a change in sort of conscious state, which certainly has to do with initiation of things and things like that, which sounds a little bit like abulia, which I think is also sort of subcortical. Are there sort of consciousness aspects that differentiate the two?
Not quite. It's a little hard to hear you on stage. Can somebody repeat the question?
He's asking about abulia.
And what about it?
Its difference from athymormia.
Abulia from athymormia. I'm not familiar with abulia. Can you describe it?
Yeah, abulia is a disorder where essentially patients seem to have a complete lack of desire, of initiation, of will to initiate, behavior, for example. So it sounds very similar to what you're describing.
Yes.
I was just wondering if there are, I'm sure there are different names, so there have to be differences, but we can probably discuss this on another time then.
Yeah, you're reminding me. So athymormia is on the scale of motivation disorders. Abulia is right before athymormia in terms of motivation. So athymormia is a complete obliteration. And abulia, sometimes they can still internally self-motivate in some cases. Of course, as you know, the problem is these are people with brain damage. You never get localized brain damage to just one region. So the disorder is involving other regions. But for the more pure cases of athymormia, I mean, they are completely lacking motivation. And there's a couple of famous cases that have been written about these. So, but they're very similar. And I think when you look at that motivation literature, you look at other things like Parkinson's and Huntington's disease as well. Really what's happening is there's a disconnection between prefrontal and basal ganglia and thalamocortical systems that are coordinating that motivation system. And it seems like athymormia is so special because you ablate both right and left hemisphere, connections to that information. So there's probably only a handful of these patients out there, but they're super fascinating, when they present.
Thank you.
Hey, so I have a question for the other panelists from Jay. I'm Brian. I work in Jay's lab. And we're doing these studies where we're looking at these like subjective effects a lot. And we've seen some really dramatic placebo effects. Sometimes accidentally, we've had a couple really awesome accidental double-blind placebos because we didn't have the equipment connected correctly, and we thought we were doing it. And we had these really, really surprising dramatic effects then sometimes on our subjects when we run the sham. And one of the patterns that I've seen is that if they've received the ultrasound prior, the placebo effect can be incredibly strong. Whereas if they're just getting sham the first time, it's about what you would expect. And I'm curious if in your own experiments, whether on label or off label, if you've seen similar dynamics emerge.
I don't know, we did not see that. On open experiments, you always assume that that there is placebo. That's why we're trying to use populations that has less of a chance of placebo, because those people do not respond to anything else. And if we see effect, I'll take it as placebo, because those people are very much suffering. So, but what we found that, and there have been studies of ultrasound, sometimes you could hear some subject kind of felt that there is vibration, or so. I think the pads that we have, we just started using those in the studies, and those the most effective way, because if there is a sound, there will be a sound. If there is a vibration, there will be the same vibration, but there will not be ultrasound in it. So just turning on and off, hoping that because it's so low in producing any side effects and people cannot really distinguish without placebo, I think you better use pads. And another thing, I have seen, I do a lot of clinical trials on other treatments for anxiety and OCD, and I've seen dramatic placebo response. I've seen seizures where we had to call the central place and break the blind and come back to the patient and say, get up. So, and complete recoveries. Yeah. Complete recoveries. So,
yeah, we had a long-term meditator that when she received a sham condition after receiving a few ultrasounds, she came back from it and said, like, oh my God, I had this physical change in my body. I had this tension in my right shoulder blade that released, and I feel physically different. And I'm like looking at her face, and there's like more blood flow and color in her face. Like I can tell she had this profound experience. And I was the one that shammed her. I didn't give her any ultrasound. What about the other? Go ahead.
Very possible. Why not?
Yeah. What about the other two panelists, the folks that are remote? Do y'all have any comments?
I can only say that my patients are unconscious for the most part, or minimally conscious. So
yeah,
the only placebo effect we can get is in the personnel, which is why we really want to blind our personnel to it. And it's an important. I mean, it's a really important issue. We do see dose effects when we do 2 sessions. We do see those effects over time. Although we have a small sample for now, so it's hard to give you a firm answer. But yeah, I mean, these are all the obvious challenges of doing this kind of research. Very important.
To just add, the effect from sham or the placebo, it's unavoidable. So when we design the experiment, we just have to do our best to make sure that it's not confounding our results. But obviously the placebo is I think it will present and it will pop up during especially the clinical trial phase, except for Martin Monti's patients.
Right. All right, well, thank you.
I think we have time for one more. I hear some rumbling stomachs, so I think it's close to lunchtime.
I have one to just follow on that comment about the placebos. Some of the reading I've done recently about predictive processing might suggest that like our beliefs and expectations have a large effect on this, like the available state space of our experiences. And so I'm curious to know if we just had a question about placebos potentially like confounding some of our results. I'm curious to know if anybody has seen like a pattern across these placebos. Is there perhaps a way to modulate them through priming and expectations and beliefs as part of the work?
Yeah, it's a great question. So he's asking about predictive coding. It's the idea that the brain is learning from past experience and using that to predict the incoming stimulus. It's sort of in this Bayesian model of learning. And I do think that the brain is doing that in some sense. And so part of what Brian is talking about, let's assume we are getting ultrasound into this network that's changing the state space and also the conscious experience that's related, we think, to that state space. The brain is constantly learning and trying to control. It's just massive control mechanisms that are learning from patterns of input, and it's using this Bayesian model. So it's trying to update how it's predicting the next input. And so the subjects know, these are conscious human beings who know, they might not know the hypothesis, but they're trying to guess it sometimes, and they have their own model about like what's happening. And so if they're able to tune in our studies to every time that ultrasound thing happens, I feel better. I have more equanimity. I'm better able to meditate. Now the whole brain, not just that network, but the whole thing is learning from that. And so part of this placebo effect could be the brain's expectation about that, which helps it change the state space, even though the external stimulus isn't coming in. Now there's an internal stimulus, if you want to think about it that way. That's a problem for well-controlled scientific studies, but that's great for clinic and meditation enhancement. So as long as we get to the effect and allow the brain to better control itself, that's really what we want to get to. And so we'd love to better characterize this sort of placebo type of state so that we could actually make it work better. So the brain sort of working with its own expectations and maybe inhibiting some of the other priors in the system that are getting in the way of that type of training.
It sounds like that's like a cliffhanger. That's
for Brian to figure out because he's asking the questions.
Very good. Thanks.
Yeah. Thank you. All right. I think we're almost out of time. Thank you so much for hanging with us all this time. Thank you to our amazing speakers, for Sasha coming in person and our folks for staying online. Beautiful talks, just such an interesting field. And you're seeing just the beginning, even though it's 100 years in the making, the technology is taking off and this is going to be an exciting place. And we hope this is a venue for some of the science to be displayed. So thank you very much.